Good evening and good morning. And we are at day 3 of our conference. Today we have 4 talks, I think, specific to the surveys. And then I have 2 talks that I think are a little bit broader in scope and maybe forward-looking, and a panel discussion. So really, without any further ado, it's my real pleasure to introduce Hiko Arawa, a longtime friend and colleague in the North Pacific. So Hiko, I'll pass it over to you.
Thank you, Brian. Hello, everyone. I am Hiko Urawa from FRA in Sapporo, Japan, and my co-authors are Shunpei Sato from FRA and Terry Beecham and Ben Sutherland from DFO Canada. Our presentation entitled Winter Distribution of Cham Salmon in Gulf of Alaska, a comparison with previous studies. Next slide. Objectives of this presentation are report stock-specific distribution and body condition of cham salmon in Gulf of Alaska during 2019 winter expedition, estimated by using new SNP baseline and compare tools with past results from winter surveys conducted by Japanese research vessel Kaiomaru in 1996, 98, and 2006. Method already reported by several authors, which were caught by surface troll of Russian research vessel Professor Kaganovsky in Gulf of Alaska between February 16 and March 18, 2019. A regional origin of individual cham salmon was estimated by using a new Pacific Rim SNP baseline developed by Pacific Biological Station. This baseline includes 545 SNPs from 400 populations, as Teddy introduced his presentation on day 1. Next slide. Left figure shows CPUE distribution of cham salmon, and right figure indicates SST map from FNA's report. A total of 223 cham salmon were caught by surface troll, and as shown here, fish were widely distributed in survey area, where SST ranged from between 5 and 7.5 degrees Celsius, which were relatively abundant in southwest water. This slide shows CPUE. This slide shows the number of catches and CPUE distribution of cham salmon by ocean edge. As seen in left figure, ocean edge 3 fish was most abundant in this survey. In right figures, almost all of ocean edge 1 and 2 fish were distributed in south of 52 degree north, while some ocean edge 3 and ocean edge 4 fish appeared in northern water. This slide indicates stock compositions of cham salmon estimated by SNP analysis. We have confirmed various populations from Asia and North America intermingle in Gulf of Alaska during winter. In the left figure, stock composition of all ages is 50% West Alaska, 5% Yukon, 6.5% Central Alaska, 11% Southeast Alaska, 9% North and Central BC, 8% South BC, 4% Washington, 20% Russia, and 22% Japanese populations. In the right figure, Ocean Age 1, Southeast Alaska and BC populations were dominant. However, Western Alaska and Yukon populations and Asian populations were not included. In Ocean 82, all regional populations appeared in Gulf of Alaska, and the South BC and Japanese populations were predominant. In Ocean 83, West Alaska, Russian, and Japanese populations were dominant, and each occupying 25%. So a proportion of Russian and Japanese populations increased with ocean age, and 40% in ocean 82, 50% in ocean 83, and over 70% in ocean 84 fish. This may be due to difference in maturing age between Asian and North American populations. This slide compares stock-specific CPUE of cham salmon in northern and southern waters. As seen here in right figure, most populations were more abundant in south of 52° north. For example, 90% of Japanese cham salmon inhabited in southern water. During 2019 expedition, we frequently found skinny cham salmon. This fish is ocean age 3 female caught in southern edge of survey area. A condition factor was only 0.756. Otoris mark evidenced this fish was released from a hatchery in eastern Hokkaido, Japan. This left figure compares condition factor and fork length of cham salmon among ocean age 1 to 4. We have defined skinny cham salmon whose condition factor is less than 0.9. As seen in this figure, many ocean 82 and ocean 83 fish showed skinny condition. And as shown in right figure, especially ocean 82 fish had a significant lower condition factor than other age groups. This slide compares frequency occurrence of skinny cham salmon among regional populations. So SNP analysis showed skinny cham salmon were originated from all regions except for West Alaska. In ocean 82 cham salmon in left figure, occurrence of skinny cham salmon was 70 to 75% in BC populations and 30 to 50% in other populations. In ocean 83 cham salmon in right figure, occurrence of skinny fish was 60% in North and Central BC populations and around 20% in Russian and Japanese populations. This figure compares stomach content index among cham, pink, coho, and Sakai salmon. Data was kindly provided from Dr. Anton Krevadorov, Russia. Thank you, Anton. As seen here, cham salmon shows lowest feeding during winter. So summary 1, the SNP analysis confirmed the Gulf of Alaska is an important winter habitat for most cham salmon populations from North America and Asia. Most of populations were distributed in south of 52 degree north, where SST ranged from 6 to 7 degrees Celsius. And ocean age 2 fish had a lower condition factor than other age groups, and 39% fish were less than 0.9 in condition factor. A SNP analysis suggested two skinny fish were originated from various regional populations, and occurrence was 70 to 75% in BC populations and 30 to 50% in other populations. Although mortality of skinny chum salmon is unknown, it may be possible that not only first winter, but also following winters are critical for survival of chum salmon. Next, move to a comparison with the previous winter surveys by a Japanese research vessel Kaiyomaru. In the right bottom corner, here are Kate Myers, John Morris, Ishida-san, and me participated in the first international Trans-Pacific Winter Survey from Tokyo to Seattle in 1996. They look young. This map shows troll survey locations conducted by Kaiyomaru between January and March 1996, 98, and 2006. Survey area included Gulf of Alaska in 96 and 2006. This figure indicates winter CPUE distribution of Ocean Edge 1 chum salmon. They are high density in western North Pacific, but rare in central North Pacific and Gulf of Alaska. Next figure shows CPUE distribution of Ocean 82 plus chum salmon. In contrast to young fish, older chum salmon are relatively abundant in central North Pacific and Gulf of Alaska. This map shows sampling locations along 145 degrees west in central Gulf of Alaska with a satellite SST image in middle February 2006. SST ranged from 4.5 degrees Celsius at the most upper station 54 degrees north to 6.5 degrees Celsius at the most lowest station 48 degrees north. This slide shows stock compositions of chum salmon by ocean age, estimated by microsatellite DNA analysis. Results are similar with 2019 estimates. Ocean 81 chum salmon did not include West Alaska populations nor Asian populations, and occupied by PWS, Southeast Alaska, and BC populations. And proportion of Asian populations increased with ocean age, and ocean age 4 fish were dominated by Russian and Japanese populations. And the left figure shows stock-specific CPUE of chum salmon by sampling location from 48 to 54 degrees north. As seen in here, Asian and Alaskan chum salmon were relatively abundant in southern water between 48 and 51. But BC populations shifted to northern water between 50 to 53 degrees north. In the right figure, estimated stock composition in entire survey area is also similar with 2019 result. This slide compares body size of Ocean 82 chum salmon caught in central Gulf of Alaska in winter of 1996, 2006, and 2019. As shown in left figure, there is no difference in fork length, and whose average is around 400 millimeter. However, mean body weight is different among years, and the condition factor was significantly low level in 2019 cham salmon. Occurrence of skinny cham salmon was 5% in 1996, 10% in 2006, and 30% in 2019. Left figure compares mean total lipid content in muscle of ocean edge 124 cham salmon captured in western North Pacific and Gulf of Alaska in winter of 2006. As seen here, in all age groups, the total lipid content was significantly lower in Gulf of Alaska than in western North Pacific. Especially a total lipid content of Ocean Age 1 cham salmon was much lower than that of older fish. In right figure, total lipid content and fork length of Ocean Age 1 cham salmon are plotted, and compared with cham salmon in western North Pacific, fish in Gulf of Alaska had a larger fork length but significantly lower lipid content. This figure compares the mean SST of pink and cham salmon habitat in western North Pacific and Gulf of Alaska in winter of 1996, 1998, 2006, and 2019. Bars indicate range of SST. Habitat SST is apparently different between western North Pacific and Gulf of Alaska, and almost 2 degrees Celsius higher in Gulf of Alaska. And the range of winter habitat SST is relatively narrow, except for 2006 pink salmon in western North Pacific. And mean habitat SST was almost stable among years. This figure compares SST and CPUE of cham salmon in Gulf of Alaska during winter and summer. As seen here, winter habitat temperatures are apparently lower than in summer. This slide shows a possible shift of cham salmon habitat in summer and winter in case of homogeneous SST increase of 1.5 degrees Celsius or 3 degrees Celsius, cited from Azumaya et al. 2007. Highest summer limit is set 15.6 degrees Celsius for summer and 7.5 degrees Celsius for winter cham salmon, according to actual observations. In summer, habitat space in North Pacific and Bering Sea is decreasing with increasing SST. In winter, the current cham salmon habitat shown in left bottom figure. Current cham salmon habitat is narrow in western North Pacific, but extensive in Gulf of Alaska. And the total habitat space is approximately 6.3 million square kilometers. In case 1, 1.5 degree Celsius increase, winter habitat shift to north in Gulf of Alaska, but it expand in western North Pacific and Bering Sea. Thus, there is almost no change in total habitat space. In case 2, 3 degrees Celsius increase, winter habitat space extremely decreases in Gulf of Alaska, but it further expands in western North Pacific and Bering Sea. So, two habitat shifts may cause strong impact on life history of cham salmon populations inhabiting especially in Gulf of Alaska. Summary 2. Ocean Edge 1 cham salmon are high density in western North Pacific, while older cham salmon are mainly distributed in Gulf of Alaska, where Asian and North American populations intermingle in winters. Most SST of winter cham salmon habitat is almost stable among years, but different by regions, approximately 2 degrees C higher in Gulf of Alaska than in western North Pacific. Ocean Edge 1 cham salmon had a lower lipid content than older fish, suggesting the depletion of their energy reserves during first winter. Cham salmon in Gulf of Alaska had a lower total trophic condition than western North Pacific, maybe due to higher habitat temperatures and low availability of prey organisms. Frequent occurrence of skinny cham salmon during 2019 expedition might be unusual event, but it could be a warning light for future salmon. So further monitoring are required to assess the trophic condition and the survival of overwintering salmon. So future climate warming may affect ocean distribution, trophic condition, and survival of winter salmon. Impact may be stronger against salmon populations inhabiting Gulf of Alaska. Thank you.
Thank you, Ross, on those striking observations. Mark, questions?
Brian, at this point, we don't have any questions. Urawa-san, I have to say that was a fantastic presentation, extremely clear, and some outstanding results. Very, very impressive. Yeah.
I wonder if
there'll be some questions.
Sorry, Mark. I'm just wondering if Christoph or Alexei are on, because I think from a lot of the material I've seen from the 2020 cruise that we didn't see a lot of poor condition chum salmon in 2020. So there might be another interesting comparison there. Christoph or Alexei, are you fellows on?
Well, we're waiting for them to chime in. Brian, there is a question from one of our Atlantic colleagues, and David Meerberg, who was of the West Coast many years ago. And are results for chum salmon available from the 2020 survey? Christoph is saying, in answer to your question, yes. So, sorry, go ahead. Go ahead, Urawa-san.
Yeah, I think stock ID of 2020 cham salmon already finished, but unfortunately, I haven't seen a result yet. So sorry.
Okay. We have a question from Lori Whitecamp. Any ideas why chum might have been skinny in 2019? They couldn't find food or question mark?
I have a hypothesis that cham salmon engage survival strategy that they overwinter less feeding than other salmon. Then energy content level prior to winter season and winter habitat temperatures may affect condition factor and the survival of cham salmon during winter. And ocean distribution of cham salmon in summer and fall are different by populations. And so I think energy content level prior to winter season and maybe winter habitat temperature affect the survival of cham salmon. So we need follow-up research in Okhotsk Sea, Bering Sea, and Gulf of Alaska during summer and fall before global winter expedition 2022. Yeah.
Good.
Mm.
Robinson, thank you very much. It is
We have one
5:25.
more. Okay,
You
there
got
was
one
one
more
more question, but if
from Jackie?
yeah, it was
Okay.
Jackie. Yeah. Do you have any great presentation? Do you have any hypothesis about why the occurrence of skinny chum was more prevalent for BC populations than for other populations?
As I mentioned, maybe trophic condition prior to winter is very important. And the ocean distribution of cham salmon in summer and fall is different by populations. And most BC populations stay in Gulf of Alaska through the year, while Japanese and some Russian populations stay in Bering Sea in summer and fall. So I suppose BC cham salmon might not be able to get enough nutrient during summer and fall of 2018 in Gulf of Alaska. That's my speculation.
Thank you. There is one
We
more
got
there,
one
Brian, but
last
I know
one there,
your
Mark.
time is tight. Yeah, there's Christoph is saying condition factor of chum was better in 2020. Question is, could the abundance of jellyfish in the north in 2019 have caused the poorer condition?
I think cham salmon prefer to feed jellyfish through the years. So I don't think jellyfish abundance affect poor condition. I don't think so. Yeah.
Brian, there's a couple more coming in. I know we're
Yeah.
out of time. I think people can still look and see those questions there. So if perhaps there can be a dialogue in the Slido response there.
Yeah, and you can certainly bring these up in the panel discussion later on too. But I think we need to move on. Thank you, Robinson. Great job.
Thank you.
And
Thank you.
Our next speaker is the revered Dr. Beamish, and he's going to tell us something new about coho salmon, apparently.
This is the ocean ecology of coho salmon in the Gulf of Alaska, and I'm Dick Beamish, and Chris Neville, Vladimir Redchenko, and Laurie Whitecamp. We put this talk together, and this is an interpretation of the results of our two expeditions, and there's more work to be done before there's some certainty in this interpretation, but I think you'll see there's a lot of new information. So we'll begin with the first slide, which is looking at the southern distribution of coho. And 2019 is a better example of the catches were in the southern part of the range of the survey, and the intensity of the purple color indicates the larger catches. And it's pretty clear that in 2019, most of the coho were caught at the southern limit of the distribution. 2020 is a little more problematic. The size of the pink circles indicate the abundance, and there is an abundance at the southern limit of the survey, but because we were not able to fish in the northern part of the survey, it's a little more problematic to conclude that coho were mostly distributed at the southern part of the distribution. Now we have one more slide showing Asian distributions. And this information comes from the chapter on coho salmon in the ocean ecology of salmon. And this is from Russian studies showing the general distribution of coho, and then showing the pink area where surveys indicated that in general, coho salmon migrate, in this case, to the south and perhaps east during the winter. However, there were Russian surveys that in the winter that did catch coho up towards the Aleutian Island. However, in general, it appears that coho salmon are farther south at the southern limit of their range. We caught more coho at night than we did during the day. And this is simply a summary of the catches in 2019 day and night. And you can see that there were 6 days that we caught coho during the day and 17 at night. And then in 2020, 8 of the 9 catches of coho were at night. And then if you look at the total catches, it's clear that we caught far more coho in the night catches than during the day. So it's possible that coho salmon are in deeper water during the day, as this works out to be 78% of the catches were at night and 90% of the abundance was at night. Now, speaking of abundance, what I'm going to show you here, let's call this intriguing, okay? The abundance estimate that was made in 2019 was 13,600,000 coho salmon, and that's in the survey area. And what I did for 2019 is I added up all of the catches in all of the fisheries for coho from Southeast Alaska to California. There wasn't any catches in California. And the total number was 2,500,000, more or less. And then using an exploitation rate of 33%, which means that the escapement would be 67%. And then an ocean mortality from after the first ocean winter, and not including fishing mortality, estimating that to be 50%, we came up with an abundance of coho salmon in the winter of 2019 of 15,104,000. And you can see that's pretty similar to the abundance that was made. And I'm sure that most people listening to this can identify a number of problems with that calculation, but I think it's also fair to say that the similarity is at least intriguing. Very quickly, a comment about predation. Laurie Whitecamp will be talking more about this, but scarring and wounding on coho salmon in both expeditions indicated that predation could be minimal in the winter. And importantly, when we looked at the results of environmental DNA, there really were few predators, particularly salmon sharks. And of course, there was some evidence of them being there, but they were very restricted in the areas that they were detected. And so it does look like there's a possibility that predation in the winter in the Gulf of Alaska is minimal. Now, this is fun. This is a scale analysis. And I looked at all of the scales that were collected in both expeditions. And the scales do not have to be from the preferred area to look at the growth beyond the winter annulus. And the winter annulus is very clear here below that orange bar. And there's 6 very clear, distinct circuli, widely spaced circuli, indicating this is a period of rapid growth. And those of you that remember about coho salmon, you'll remember that coho salmon in their second ocean winter are the fastest growing of all of the salmon. So in looking at the scales that were collected, most fish in February and up to mid-March really had not started this period of rapid growth. But fish that were caught in 2020, towards the end of March and in April, you could see the beginning of the period of rapid growth. And this is relevant for the next slide, which relates to the diets. Now, in 2019, the diet indicated that the dominant organism were pteropods, 73%, and there were a few other organisms. And what we are concluding is that we're calling this a maintenance diet. This was a diet that basically allowed for metabolic processes, but with really no growth, as indicated by the scales. And it wasn't until later on in March or April that we started to see more rapid growth. And that in 2020, that corresponded with the diet of, now I'm going to pronounce this Ocutania, which is a very interesting squid that apparently is starting to increase in abundance this time of the year and in the south, which also may partially explain why coho are in the south. So it appears that the diet of squid, I said was related, but it perhaps could be related to the beginning of rapid growth in coho salmon. The next few slides are nothing but spectacular. This is the composition of the coho catch in set 4 with a little bit of set 5. And those of you that know anything about the expedition in 2020 know that in set 4, so shortly after the expedition started, we got a large, well, relatively large catch of coho. There was a few in set number 5, but importantly, when we came back to this area 2 weeks later, there was no coho there at all. So if you look at the DNA, we've looked at 52 so far. And if you look at the composition of the DNA, basically it includes stock from southern US right up to Alaska. And there's even one fish that was identified as coming from Alaska. That may be a baseline problem, and we'll work on that. But what we saw in this one population, or the catch from that population, was a stock composition from populations basically throughout the range of coho salmon. This slide is a lot could be said about this. I'm sure that most people have never seen this before, although it has been published. And what this is in February of 2010, we had a survey for salmon in the Strait of Georgia. And in one of our 30-minute catches with our standard trawl, the catch was, now the estimate was 6,000 coho, almost impossible to believe. So obviously they didn't count the coho, but certainly thousands of coho. Now, a few things about this. This large school we're interpreting this that they came in and left. There was, in the recreational fishery in March, April, and May in 2010, there was no coho caught. So we're interpreting that to mean this was a school that entered the Strait of Georgia. The composition within this large school is mostly coho from populations around the Strait of Georgia and Puget Sound. And our interpretation is that, and you'll see shortly, that this time in these years, coho would leave Puget Sound and the Strait of Georgia and go to the southwest tip of Vancouver Island. And what we're concluding is that in some years and sometimes that those populations actually formed this very large school. And it was that large school that entered the Strait of Georgia, didn't find food according to our interpretation, and turned around and left. And then one more observation about schools, and a really important observation from the 2019 expedition. And this is a camera that was on the net, and the camera identified 10 coho that had entered the trawl net. You can see the shadows of them there in the orange circles. Of those 10 coho, there was only 3 that were eventually retained, so 7 escaped. But importantly, these coho were only in this area in the net for estimated about 7 minutes, or about 12% of the trawl. So there was a brief period during the trawl when an abundance of coho entered the net, and with 2/3 of them escaping. So pretty good evidence that schools of coho exist in the winter. Now, we're not saying that this happens to all coho, but it's certainly an indication that schools do occur. They can be really large, and they can be composed of populations from all up and down the coast. I think quite a spectacular discovery. Now, the next few slides are summaries of some studies that were done. We'll just call them the Welsh-Trudell-Morris studies. And these individuals carried on surveys on the West Coast, actually from Washington Strait up to Southeast Alaska, and February and March, and really from 1995 through to 2011. So this first paper is looking at only hatchery fish, only fish with coded wire tags. And over this 10-year period, 98 hatchery fish were caught in the survey, and you can see 94 were from the Strait of Georgia or Puget Sound. But really, again, importantly, there was no coho with a coded wire tag that were found in all of the other survey areas. Now, what we're not showing here is that they also had surveys in the fall. So the stock composition, now remember, these are just hatchery fish that existed off the West Coast in the fall. Those fish all left that area and presumably moved out into the open areas of the Gulf of Alaska, and they were replaced by coho salmon from the Strait of Georgia and Puget Sound. Now, the same investigators, now these are some of the same data. What we did here, I don't think these data are published as one overall summary. We took the individual catches from the various cruise reports that were put together by John Morris. The numbers are close, they may not be exact. And so over this period of 11 years, this time, now this is just the winter again, so 950 coho salmon were caught in the winter months on the west coast of Vancouver Island. Some were caught in inlets to the north of northern Vancouver Island, but again, they were caught in the area off the west, the outside tip of Vancouver Island. Those would be Puget Sound, Strait of Georgia. So there were some catches from the inlets. Now the summary is that, again, virtually no coho were found in the winter off the West Coast over all these years from the tip of Vancouver Island into Southeast Alaska. And repeating, small abundances were resident in some inlets off the west coast of Vancouver Island. And these included Strait of Georgia and Puget Sound coho. But mainly, large abundances of coho leave the coastal areas and should be in the survey areas in the winter. Now, this is a summary of all the DNA that we have so far. So a summary of 215 fish individuals. And these include the fish that were caught in the school, all right? And not in the school in the Strait of Georgia, by the way. So these are just the fish caught in the 2 expeditions. And so the summary is that coho from the southern US populations remained in the south. The Strait of Georgia and Puget Sound populations really do not move out into the open ocean, but remain close to shore on the west coast of the southwest coast of Vancouver Island. Central British Columbia populations move off the shelf and are distributed throughout the survey area, again with concentrations in the south, as we said at the beginning, that coho tend to be concentrated at the southern part of the surveys. Northern British Columbia populations were found only in the southern areas. And interestingly, Alaska populations were distributed throughout the survey area, again with a concentration in the south. And just to repeat the information from the schools again, the set 4 with a little bit of from set 5, those populations were from all areas off the coast of from southern US to Alaska, and there was that Russian fish there. And then just to repeat, the 2010 school was mostly Strait of Georgia and Puget Sound populations. So a summary then, what is it that we have learned? And I would add, what have we learned that is new? We learned that coho salmon in general are found at the southern area of their distribution in the winter, and they may be in deeper water during the day, and that would be in the winter. That may not be the case outside of the winter. Large aggregations of populations may form in January, February, and March, and they are not associated with other species of salmon. And they appear to be feeding mainly to maintain metabolic requirements with minimal growth. Now, if there were abundant prey during these periods, it's certainly possible that rapid growth might start. Most populations move off the shelf in the fall, and they are distributed throughout the survey area, again with concentrations in the south. Some populations remain in the coastal areas. Predation mortality in the winter may be minimal. I think that's important. And squid abundance may be associated with the beginning of rapid growth, and that would be late in March or early April. And that's the end of our presentation. Thank you.
Thanks, Dick. Lots to think about there. And that, I see Mark has gone dark, so maybe he's not around. So he must be having a problem.
No,
there he is.
No, I'm good. I'm good. Sorry about that.
So.
Yeah, I'm not seeing any questions come in yet. But Dick, I'd be interested in your thought that predation might be low. If you know, I can't remember what the rate Laurie said of the scarring, but if we don't know how many are actually dying from that scarring, and some of these predators aren't available to the trawl, what are your thoughts on why you would think that the predation is low?
Because we don't find them. It's not that predation. Now, Laurie did explain it quite well. The concept that predators are a major source of mortality, in all fairness, has always been made up. And what we are finding, I think, in both expeditions, and the eDNA is also, I think, really helpful, is that the predators simply aren't there. So, and you know, as I said, we never really knew that they were there before. We just had no other way of rationalizing what would kill fish. So we just naturally said predators. And I'll tell you that Chris and I sometimes think that predation is maybe a cause of death, but not really a regulator of abundance. And actually, we're working on whether or not we can in some way write about that.
Okay, thank you. Lori says the rate of scarring was 2%.
Yeah.
A question from Will Duguid. Interesting talk. What role could patchiness play in the apparent absence of coho from some regions in winter? I.e., with 6,000 fish in one set, the density would be high, and the schools presumably would be relatively small in area, and therefore rarely encountered.
Well, first of all, the existence of schools almost certainly is occurring. Whether or not all of the coho occur in schools, I doubt that. So patchiness, in the sense that you might normally see that as something that would occur with, say, juveniles when you're doing juvenile surveys. But the concept of large abundances and for coho in the first ocean, well, it's only ocean winter. Yes, that obviously that would be patchy in the sense that those we don't know how many there would be in a school, but that would affect the trawl survey. But then go back to that estimate that was made on total abundances. I mean, again, it's problematic with the calculations, but the estimates do seem to be reasonable anyhow. So not able to answer the question about what patchiness means in terms of the overall distribution of coho, other than it certainly occurs. How long those schools retain, it's, they could be in a school for several months. Chris and I think that those schools are probably the fish that are in schools might stay until they find prey abundances such as squid.
Okay, we've got a question from Jackie here. Says, would you change any of your conclusions or new things learned if 2019 and 20 MHW conditions impacted coho prey availability? For example, lower prey abundance than in years not observed. And I'm sorry, I'm not picking up on what MHW
That's probably
conditions are.
marine heat wave or something like that.
Oh, yes, that'd be it. Yeah.
Yeah, but of course that would be the case. In other words, there both 2019 and 2020, the prey availability could be minimal. I think that squid are the major prey that coho are looking for, but certainly that could be the case. In other words, in 2022, if the oceanography was more normal, we may not see any evidence of schools.
Okay.
Okay, Mark, we're into our break time here.
Oh, okay.
That, you've got one from Jacques, I see.
Yeah.
Jacques, I wonder if you could keep that. It's a good question for the general discussion later on. And so if we can park that one, we'll try to get back to it because I think it is an issue we have to address. All right, we are into your break time and we are back at 6 p.m. Pacific time to hear about the energy density work by Charlie and others. All right, so thanks, Dick, and we're in a break. Okay, welcome back. We're all refreshed and ready to go. So, Charlie, if you're ready, let's move on. Todd Miller is presenting. I'm sorry, I didn't have that.
Okay, great. Thank you. My name is Todd Miller, and I'm from the Alaska Fisheries Science Center for NOAA Fisheries at Auk Bay Laboratories in Juneau, Alaska. And so basically today I'm going to present some of the results from the 2019 survey that involved energy density measurements, lipids, and then some of the stable isotope results that kind of show some of the trophic overlap or niche overlap. Okay. Phone went asleep. Okay, here we go. Okay, just getting set here. My picture is blocking part of the screen. Okay, got it. Okay, so for winter as a critical period, we all know that that hatchery production has increased in over 50 years dramatically, and that the winter time is kind of this critical period where prey availability is rather reduced. This large input of hatchery produced fish can produce both inter and interest competition. And at the winter periods, it's possible that this level of competition may be highest during that time. And the thought about this is that this may lead to highest mortalities in the first winter at sea. So that's the general question. So the aim and objectives of the study, the aim was basically to better understand the marine ecology of Pacific salmon in the critical winter period. The objectives were to assess winter fitness through energy density, lipid, protein content, and then to assess trophic overlap amongst salmonids. And then determine the hatchery component of the pink and chum salmon from the fish that were collected to assess competition and whether hatchery versus non-hatchery are competing. And then a last part of our component is to conduct bioenergetics modeling to assess salmon growth rate potential in the Gulf of Alaska in winter. However, we will not be presenting results from that at this time. That's still in progress. Okay, a summary of results. I think we've all seen this many times before. Basically that from 2019, we collected mostly chum, followed by coho, sockeye, and pink. We performed energy density measurements using bomb calorimetry, lipids using microcolorimetric method called SPV for the abbreviation, and protein through proximate composition. And then we ran bulk stable isotopes on mussel plugs from all the species, except for Chinook. So looking at the results from the effects of length on energy density, we see that there is a significant, although slight trend in chum and sockeye salmon. This trend we would expect to see a lot, the slope being a lot steeper during summer months. So this kind of flattened or much more flattened is not surprising if they're not growing a lot during wintertime. And we can see that coho and pink are not showing any significant relationships. And in fact, they're very flat, and pink is almost, as a general trend, is decreasing with size, although the size range isn't that big. There's a general trend there. And we'll see with the proteins and protein and lipid analyses a little bit more about the condition of all these species, but the one with regard to pink might be indicative of poor nutritional condition. So looking at chum salmon by ocean age, we have ocean age 1 showing a general decrease. None of these, as you can see, these relationships were significant. However, there are general trends there. ocean age 1 is just showing a slight decrease. Age 2 flattens out. And then starting age 3 and 4, we're starting to see this kind of increase with size. So it's possible that ocean age 1 and 2 are kind of struggling, and 3 and 4 are just kind of in better nutritional condition. So looking at, one second here, I've got this screen in the way. Looking at chum with respect to the thermal marks, those that it's really ocean age 1 that we had enough to run any kind of trend line through it. However, none of these were significant or of anything that we could run. But what you can see is that the thermal marks and the non-thermal marks are showing a general decreasing trend in ocean age 1. So they're both kind of trending in the same direction. When we look at sockeye, the age 1s are, well, there's just not enough data to really say anything, but in age 2 and 3, there's still not a significant relationship. They are rather flat, and so this kind of makes us think that it could possibly be that that they're not doing that well nutritionally. So if we look at hypothetical scenarios of nutritional status, so we're looking at fit, not starving, and starving. And this is to generally look at our lipid and protein results from the 4 different species. Is that a fit individual or a fit group will have a positive relationship between lipid and protein and weight. Not starving, you generally do not have any lipids that are increasing with size. However, you could. And then starving could have a flat, no relationship, or maybe even a negative relationship with lipid and protein and weight, where you have a condition of catabolism and the fish is essentially feeding off its own proteins. So looking at lipid and protein by age class for chum, we see if we first look at percent lipid and ocean age 1 and 2s, these are very low lipid levels. I'm not sure if these are low for those age classes. That might be a question for someone out there, but they are well under 1%, so they're really low. And so we're not really seeing any kind of definitive trends there. The only one interesting thing is that if you look at the thermally marked fish, they are showing a positive trend versus the non-thermally marked fish. So our hatchery fish feeding better or kind of competing with the wild, quote unquote, wild fish and causing them to decline. So we're not really sure. These are preliminary numbers. But and then as we go, as you look at ocean age 1 and 2, well, all of them on the percent protein, we're seeing that strong positive relationship. And we're in percent lipid for ocean age 3 and 4, we're seeing that positive relationship. So you could generally classify this as the at the earlier years, it's not fit, but then as you progress to ocean age 3 and 4, could be getting more into a fit condition. When we look at sockeye, again, we have some trends in ocean age 1, but with the small sample sizes, it's really hard to say. But as we get to ocean age 2 and 3, we're seeing actually kind of a flat, an increasing trend in percent lipid, but the percent protein is rather flat or even decreasing. So this would not be necessarily indicative of a really good nutritional condition, certainly not fit. Lipid and protein for coho and pinks. If you remember, I mentioned earlier about looking back at with the energy density and looking at COA and pinks and how they weren't really showing any kind of trend with energy density. You can see the percent lipid of coho is decreasing with size, and it's pretty flat in the percent protein level. It's a slight increase. But when you look at pink's nutritional condition or the trends are really showing kind of very poor condition. We have reduced protein levels and reduced lipids as we're increasing in size. So generally pinks do not appear to be doing that well. Okay, I mentioned earlier we did some bulk stable isotope analysis, and these are some of the results of looking at the trophic overlap and how that might kind of fill in some of the gaps in terms of competition between salmon species. And the initial look at all the stable isotope values of all the different species, it was really obvious that chum salmon had like this extremely protracted range of isotope values that were beyond any fish could really have. They'd be, if they had that, they'd be like the super generalist and the best competitor. So, but it was pretty obvious. And so, we looked at it with respect to size and ocean age, and it teased out this one group, the ocean age 1, and those clearly showed a coastal signal. They had very high carbon and nitrogen isotope ratios relative to the other species. And so that's really pretty much indicative of that coastal signal. Now, you'll see trend lines in there. Some of these, they seem to be just, they're going up, they're positive slopes. I'm not sure if that's their ontogenetic shift, isotopic shift by higher trophic level feeding, but there's something going on there with that. Now, I guess the big question is with the chum that ocean age 1 is, are they recent arrivals to this area and they have that coastal signature just because they recently arrived? Or the other scenario is that they arrived in the fall or sometime after then, and they just didn't grow very much. And added tissue growth is one of the biggest ways isotopes or an organism can reach its isotopic equilibrium. So this is a question that's really interesting. I think. Well, one of the things that we'd like to do, or we plan to do, is to look at the islands of different species and pick out certain species, look at the islands and do isotope analysis on the layers of the lens. And we hopefully will be able to come up with some kind of isotopic trajectory that's indicative of their movement from the point of origin to their capture. And this would be really cool if we could also link it with genetics and look at stock origin and kind of come up with some isotopic trajectories based on that. It is a lot of work. One eyeball can be as much as like 30 samples. So it's a lot of work to do, but something we're very interested in. We did this also we saw this also with sockeye salmon, but not as clear and a little bit different trend, but definitely like this movement from a coastal signal to an offshore signal. The trend lines within ocean ages were opposite. They're decreasing. So I'm not really sure what that means. If they've equilibrated out in the open ocean, they're showing a decreasing trend with size, then that would suggest that they're feeding lower trophically as they're getting bigger. But one other comment I wanted to mention is that, but it's definitely like the carbon signal is clearly showing that it's moving from this coastal oceanic. So it's yeah, there's something going on here, both trophically and movement-wise. So we took that kind of information, helped us guide us through doing a stable isotope Bayesian ellipsis in our model called CYBER, where you can build prediction ellipses of 95% of the data within a species that represents the isotopic niche space. In this case, I removed the chum age class because, like I said before, it would have made chum like cover most of the species, and we didn't want to contaminate the oceanic signal with lingering terrestrial signals. So, and then I used what's called niche rover to come up with the estimate of niche overlap. It's a probability of a species A that would land within the niche of species B. So it does these comparisons. And the results of that showed were pretty interesting. Chum and sockeye showed the greatest overlap, not very surprising there. But what's really interesting is that pink salmon showed the highest overlap across all species, with generally an average of around 70%. So pink can represent a a level of direct competition or resource competition with these other species, more so than any of the other salmon. At least that's what this is suggesting, that they can directly compete more for food across more species. So that's it. The next steps, we want to analyze 2020 samples, look at eye lenses, do more energy density and lipid analyses. We want to incorporate more IYS data, incorporate data from our juvenile surveys if we actually get some. And then we'll move forward with the bioenergetics modeling. And acknowledgements, thanks to everyone's sample collection, sample processing. Thanks to Chris for the data. Any questions? Yeah, you can ask Charlie Waters, or you can contact me at todd.miller@noaa.gov, and I'll respond. That's it.
Thank you, Todd. And that some very interesting observations there. And that while Mark is waiting for our questions coming in, what do you guys think is going on with the coastal signature for chum when we're sampling in the wintertime? And that, or I guess the question would come down to how quickly does that signature change?
Yeah, that's really, I mean, that's one of the problems in isotopes is that, we hope that they're all like equilibrated within the same ecosystem or same location. That's almost always not the case. Although, it's very interesting to see how different the H0 chum are. So it'd be very interesting to tease that out.
We don't have a question coming in yet, but what about with the energy density and the skinny chum? Did you look at the skinny chum at all? Maybe you didn't have access to that?
I may have. That was from 2020. Is that correct?
No, I thought it was 19.
Oh, it was 19.
Yeah.
I did not run those analyses. I can check on that, but they might have been some of our low chum values. I'm not really sure.
Mm-hmm.
We should definitely give you the sample numbers on that so you can check that out, Todd. Go ahead, Mark.
Oh, yeah, that would be great.
Question from Francis Juanez. Did you try to estimate mean trophic level within and across species?
Yeah, thanks, Francis. Yeah, this would really be great, is to actually come up with a trophic level. We didn't really measure many prey that I would feel confident in saying, using it as a nitrogen baseline to set that. Although Brian Hunt probably has measured a lot of that and would be able to establish trophic levels. We didn't really have plans to do compound-specific stable isotope analysis where with nitrogen we could establish trophic level. But yeah, Francis, that would be very helpful to tease out. And one way to do that is just take a subsample, maybe run compound-specific on it, and use those localized range of nitrogen as anchor points to set trophic level for everyone else. So just need time and money.
Yeah, I'm not seeing anything else, Brian.
Okay, well, another one that comes to mind then, Todd, and thinking future-wise, are there samples of the food items that we should acquire that would help you with some of the signatures you're just talking about? I'm just thinking, should we be collecting some specific samples for you and treat them in any specific way?
Yeah, well, I think, that's a very good question. I think, again, Brian's doing a lot of the kind of full food web analyses, and his sample collections cover they cover a broad area and taxonomic groups. So he would really be, his analyses are really important in that regard. We did collect some. I believe we have euphausids and some other, but really, it's your view of what an organism is doing trophically is only as good as your collections and your analyses. So if you only have euphausids and maybe myctophids, and then you throw it up there, the models will give you proportions, but only relative to those two other prey or three other prey. So we look forward to working with the rest of the group on trying to plan collections and support the stable isotope results.
Evgeny notes that it's been, assuming this is some of the prey items, it has been collected extensively during 2019 and 2020.
I'm sorry,
Evgeny's
responding. Yeah.
Sorry, Evgeny came back with a comment to say it has been collected extensively during 2019 and 2020. So I'm assuming that's talking about some of the items that you were suggesting we needed to be collecting. Prey items, yeah.
Right. Okay.
Yep, that's it.
So I suppose if Evgeny is listening, have those samples being processed, or are they being held that we should get them to Alaska for processing?
I think we have some samples that we were able to bring back. 2020 is and 2019. I'm not sure about 2019. Some of the most of that came back, but with COVID in 2020, it's just been.
20, yeah.
I mean, I could meet you at the border and we could throw samples across, maybe, but yeah, that would probably we may have to wait for that.
Yeah, we tried that with we couldn't throw enough.
So another comment from Evgeny saying, yes, and Brian will be presenting them next. So
Good.
I think we can stay tuned for Brian's.
Good. Well, thank you very much, Todd. Nothing else there, Mark? Because we're pretty much on track here. And so we're in 1 minute. So the next speaker is going to be Brian Hunt. And he will be talking about the food web studies and diets. So as we get ready to go, I think we're close enough. We can just give it to Brian.
I hope that you can all hear me okay. Yeah, this is good timing. So Todd, that was great to see your presentation, and I think that I can answer some of the questions here. So I'll be presenting just an overview of our North Pacific salmon food webs. North Pacific salmon food webs work, and you can see there's a few collaborators and quite a lot more that are not listed. Next slide. So just a quick outline. I'm going to just elaborate on the objectives a little bit, and then I'm going to give a little bit of details on the approaches that we're using to measure food web linkages, and those are the main methods are stabilized isotopes and fatty acids, but also using gut content analysis. I'll show you a bit of preliminary data, and some of that will speak to some of the questions that you've had, and then I will get into some next steps. So next slide. Next slide, please. So the overarching objective is to, very broadly, to resolve the feeding ecology of Pacific salmon and the structure of the food webs that support them. Next slide. And there are three sort of overarching questions here. So one is, what is the North Pacific Food Web Network? So here our objective is really to provide a framework of connectivity between components of the North Pacific Food Web from phytoplankton through to the highest trophic level that we can sample. And that this will be something that one of the outputs of this will be a framework for ecosystem modeling. Very specifically, we want to know where do salmon fit within this food web network, and this will touch on the topics of the winter foraging ecology, the trophic niche, and competition between species. So some similarities and collaborations to come with TOD, that's for sure. And then how does foraging ecology impact health? And so we've seen a few different measures of health already with energy density, lipid content, protein content. And we're bringing an additional method, and that's the fatty acid analysis, and really looking at how food quality, so establishing food quality and relating this to salmon nutrition, so understanding salmon health. Next slide, please. Okay, so I'm going to give you a little bit of background here. So some of the things that are useful to know for these dietary studies is that we'll have a fish that consumes various things. So it is ingesting them. This stuff goes into the gut, and we can catch that fish and we can see what's in the gut. And that's a snapshot in time. So the diet analysis is giving you a snapshot of a few hours in the life of the fish and what it was feeding on. Some of what is ingested is actually assimilated, and so that becomes the tissues of the fish, and then some of that is digested. Next slide, please. And for the ingested work, yes, we can, like what's in the gut, we can use microscopy, those gut content analyses. Genetics is also a really good tool for this that we've been starting to use a little bit more. And then for the assimilated diet and what's actually retained by the fish, the stable isotopes and fatty acid are great methods for investigating this. And then the ingested stuff we don't really get into at this stage. Next slide, please. And so, yeah, let's just take a quick look at the carbon and nitrogen stable isotopes, because I think it's usually the case that not everyone is familiar with this. And so, yeah, our focus is on these two aspects, or these two components, carbon and nitrogen, and each of those has got two isotopic forms. There's a light and a heavy form, and so these isotopes exist naturally in ratios in the environment, and those are taken up by the phytoplankton and then transferred through the food web, through the tissues of organisms, successively through the food web. And one of the things that happens in this transfer is that the heavy isotope tends to accumulate. Well, it does predictably, and the lighter isotopes are more are preferentially metabolized. And so you have this accumulation of heavy isotopes as you move up the food chain. And because there's a predictable fractionation from one trophic level to the next, this gives us a lot of power for interpreting food web dynamics. And so the fractionation factor for carbon is around 1, and for nitrogen, it's around 3.5. Next slide. And so if we can measure all of the components, so we can take samples of everything in the system and measure the isotope values, we can map them out in isotope space, And in this diagram, in this conceptual, well, it's not, yeah, I guess it's conceptual food web model on the left-hand side of the screen. The animals are distributed according to nitrogen on the y-axis and carbon on the x-axis. And you can use the carbon and nitrogen values to trace the passage of materials through the food web. And so, yeah, there are three key aspects that we can derive from the isotopes, or three that we're interested in here. One is to estimate the trophic level. So we normally do this using the nitrogen isotope value. We can use the carbon to trace organic matter sources. And in this example ecosystem on the slide here, you can see that there's very different. If you look right down at the bottom, there's quite a range of carbon isotope values for phytoplankton on the one side, and then various other macrophytes on the other side. And so, yeah, you can use the carbon isotopes to trace the inputs of these autotrophic contributors to the food web. And then we can combine the carbon and nitrogen to also to estimate trophic niche width and overlap. And one of the things that's important to know about the isotopes is that they're a time integrated value. And for salmon, for adult salmon, this is we estimate this to be on the order of 4 to 9 month integration. So what we measure when we go out there in winter, well, in March, is reflecting consumption over many months before that, back into the fall, probably. All right, next slide, please. So there's some other things that are really useful with isotopes, and this gets a little bit onto what Todd was talking about with the chum salmon having this elevated coastal signature. So one of the things we have done is we've developed isoscapes for the North Pacific using zooplankton data. And so we have predictive models that allow us to estimate what the trophic baseline is across the eastern North Pacific. And if you look at these two figures on the left-hand side, we have carbon on the top and nitrogen on the bottom. What you can see is that there are elevated values of carbon and nitrogen in the coastal waters, the shelf waters, and this is because of higher productivity. So the main driver here is nitrates and drawdown by the phytoplankton. And so this is really useful. So we can predict what the isotopic values are across the Northeast Pacific, and that allows us to then assign a location to fish if we measure their isotope values. And it also gives us an index of regional productivity. So we can, based on the isotopic values, if we know what the trophic level of the fish is, we can estimate the productivity of the system that they came from relatively. Next slide, please. Okay, so that's the isotopes. And then the fatty acid side. Fatty acids are really useful as well. They can be used as trophic markers, and they can also be used as indices of food quality. And so how are they used as trophic markers? Well, there are essential fatty acids that are not produced in great quantities by heterotrophs. So they're produced by autotrophic organisms, and that's how they are generated and delivered to the food web. And different autotrophs produce more certain essential fatty acids in more or less quantities. So it's quite diagnostic. So we can use those fatty acids to estimate contributions of different autotrophic sources into the food webs, and that helps us understand the dynamics of the lower part of the food chain. So particularly connectivity between phytoplankton, bacteria, microbes, and zooplankton. So really the foundational stuff that sets productivity in the system. And then we know, everyone knows about omega-3 fatty acids, very important for health. And so there's various sort of indices of health that can be derived from fatty acids. And one of those is the DHA-EPA ratio. And it's generally considered that a DHA-EPA ratio of greater than 2 is optimal for fish growth and performance. So that's a metric that people have often used, and it's used quite a lot in aquaculture. Next slide, please. And this is really relevant because prey quality, and using DHA-EPA as an example, is very variable. So not everything has the same value. And it's not just about total lipids or total proteins. It's also about the type of this lipid, so the type of fatty acid. So that's something that's very important. And so this figure here is showing you DHA EPA ratios measured for a suite of planktonic organisms from the Strait of Georgia. And you can see that there's a really big range. They go from near zero across to just over 2. So the highest DHA EPA ratios are recorded in fish larvae. And so this is very interesting because it's telling you that the prey quality is very variable, and so we should really be paying attention to what the salmon are eating. And most of the zooplankton, interestingly, in the Strait of Georgia, for this study, this is summertime values, they had DHA EPA ratios less than that optimal value of 2. So we want to go out to we want to measure this in the Gulf of Alaska and beyond across the Pacific. We also don't have data for squid and myctophids, and so it's potentially important forage species. So we want to establish this. And also to measure the total fatty acids and generate different metrics of food quality using the fatty acid approach. Next slide, please. Okay, so what we've done so far for 2019 and 2020 is that we have sampled everything we possibly could in the food web. So we have collected particulate organic matter. We have using the Niskin bottles that you can see on the top right. So from water samples, we had to use bongo nets to sample the mesozooplankton, and we have full samples of zooplankton by size fraction and also by major species for the macroplankton. And then we've also sampled the trawl net as much as we can to try and capture from the smallest to the largest organisms. And we've taken samples that we have then analyzed for stable isotopes and fatty acids. I will say the one area that we're lacking in, our weak points, and all of our weak points in this analysis or in our survey so far is what's between the zooplankton and really the micronects and the macrozooplankton. So a lot of the organisms that the fish were eating, like the pteropods being consumed by the coho, we didn't catch this. Cleopyramidata, we didn't catch the species in any big numbers. We were just missing them. We didn't have the right gears for it. Next slide, please. Okay, so this is our workflow. For example, we get the trawl, and then we take representative samples of everything that we can. We collect tissue samples after our species IDs. We also take length and weight data because the size of organisms is obviously very important for foraging ecology. And the stable isotope samples are just frozen at -20 on board, and then we get them back to the lab. We have processed, we've done a lot of processing already, and we use those stable isotope values to determine trophic level, measure food chain length, identify salmon trophic niche, and also, as I mentioned earlier, use it as a productivity index. The fatty acid samples need to be treated a little bit more carefully, they need to be frozen at -80. And we use these to identify primary producers, the nutritional quality of prey types, and also the salmon nutritional health. Next slide, please. Okay, so where are we at so far? So we processed everything from 2019, about 50% of our samples from 2020. We have about 2,000 samples from 2020, so there's a lot there. The fatty acids, this is coming. We have done a lot of the preparation work. We haven't done all the analysis yet. And this is in large part because this work has been designated for a postdoctoral fellow who will be really focusing on these data and supported by the BC Salmon Restoration and Innovation Fund. Okay, so that means that like the full-blown analysis of these data are still to come, but I want to give you some preliminary results, and I've already shown you some at the Pisces meeting back in November. But let me show you some results. Next slide, please. So this is a very simple, just an isotope biplot. It's showing you the nitrogen isotopes on the y-axis and the carbon isotopes on the x-axis, and this is for our salmon species from 2019 and 2020. And so I have a little arrow there indicating increasing trophic levels. So that's usually in the y-axis direction. That's what we use to estimate trophic level. And so touched on this phenomenon of the higher isotopic values in shelf waters, and I demonstrated that with the isoscapes. This is a normal part of this is because of the productivity in the system. And I gave a presentation on this at Pisces last year about this exact thing. So what we've done is that we have we have taken into account this coastal signature in the offshore fish and to standardize our values when we estimate trophic level. And I'm going to show you some trophic levels shortly. And for these plots, I haven't included any species from the shelf. So what you can see here is that we have a very similar pattern across years, and it's interesting that we have chum that's got the lowest values for carbon and nitrogen and the highest values are for coho. We didn't, if you recall from Todd's last slide for 2019, we only had 3 Chinook, so there weren't a lot of fish there. And so let's go to the next slide. And so if we calculate the trophic levels, and I've done this here just for 2019, so this is trophic level corrected for that shelf signature, you can see them in the trophic levels in the table on the bottom right. So highest trophic level is actually the coho at 3.8, right up there with the Chinook at 3.7. This is not taking into account size differences. And we had one huge Chinook and we had a couple of small ones, so that bigger fish had a higher trophic level. Pink and sockeye, the same trophic level, 3.6, and chum at 3.2. So that's pretty interesting. That's quite a range. So let's go to the next slide, please. I'm going to come back to those trophic levels, but I wanted to show you. This looks like a mess, but yesterday there was quite a lot of discussion about the micronectin and whether they're prey or actually potentially competitors. And so I just pulled out some of the core sort of micronectin species, and so we have some squid and some ectopids in there with the sockeye, pink, and coho and chum. I didn't include Chinook. And I threw in 2 zooplankton species that are important. They are like good dietary items, the sort of Neocalanus and the Euphorsia down on the bottom left-hand side. And so what you can see, not very clearly, but I mean, it's kind of apparent in this mess is that there's quite a lot of overlap between all of those groups, and that's telling you something. That's telling you that they have very similar isotopic space. So there is certainly some overlap in the resources that are making up their body mass. So there's this, looking at the notes on the side there, there's this isotopic overlap between the salmon and topids and squid. But I will note that the size is really important. So you can see that there's some quite big error bars, and that's like, for instance, the Boreateuthis. We have small ones and we have big ones, and they're very different trophically. So the bigger squid are certainly more, they have higher trophic level than the smaller ones, for instance. Right, let's go to the next slide. Okay, so coming back to trophic levels now, we can put the salmon in a plot of trophic level for some of the main groups. We have everything resolved by species, but I've mostly compressed those species into groups. And so I've highlighted the salmon in red boxes, and then you can see the rest of the organisms. And if you can see myctophids right there having a similar trophic level to the salmon, the squid as well. The squid also showing that wide range. There were actually a couple of squid species that had very low trophic levels, and there were others that had quite high trophic levels. It wasn't just a size thing; it was also a species difference. Okay, so this is just demonstrating again that the salmon trophic level overlaps with myctophids and squid. So let's go to the next slide. And so we can plot these different organisms out on the food chain. So we expect phytoplankton to be trophic level 1. We had a couple of zooplankton species that were actually at trophic level 2 based on our calculations. So this included tunicates. So there were a lot of salps out there. And then also this other pteropod, Limacina helicina, that's indicated on the plot on the figure here. And then Neocalanus, so the copepod that's really important as a forage item, and Euphorsia pacifica. These had quite these had higher trophic levels. They certainly had phytoplankton in their diet, but they were also eating other zooplankton. So they were certainly omnivorous. And then if you looked at the myctophids and the squid and the salmon, there was actually quite a lot of overlap. So the isotope data indicate that the zooplankton are consistently a major portion of the diets of salmon. If you look at the trophic levels, they're sort of mid-range between trophic level 3. And this is what we saw in the foraging, that there were certainly in 2019 there were a lot of krill, pteropods in the diets. In the salmon species, there is some separation among the species, but particularly chum. So in terms of, certainly from a trophic level perspective, the chum at the lowest trophic level, and that seemed to match, what we know about chum is that they have a higher gelatinous component to the diet. And that salt, the tunicate, the salt that we saw, that was very prevalent in 2019, this had a trophic level that would generate that sort of trophic level in chum, if chum would be on them. And I think one of the important sort of indications from the isotope data is that we do need to consider micronectin as competitors, not just as prey. Next slide, please. I just wanted to put this in because we did, Chris alluded to, or showed some data actually from these Western Pacific samples that our Russian colleagues managed to collect for us. And we don't have a lot of samples, we have a few. And so I'm showing you the isotope data for those. So I'm just showing you sockeye salmon. And so one of the things you can see on the map is, so the color coding for those points is indicating red is a higher nitrogen isotope and blue is the lower isotope, nitrogen isotope value. And you can see that those Western Pacific salmon all had more isotope values that were on the higher end of the spectrum, whereas they were relatively lower in the Gulf of Alaska. And these higher values indicate either higher productivity in the West, which we know to be the case, there is higher productivity there, and or feeding at a higher trophic level. And so what we hope is that with the collections across the Pacific in 2022, sorry, not 2020, we will be able to resolve this. And just one other interesting observation, in comparing those Western Pacific samples with the Gulf of Alaska, if we compare sockeye and pink, so this is this plot on the right-hand side. Sockeye are in blue, pink are in pink, and the triangle is the west, and the circle is the east. And so if you look at sockeye, there's both sockeye and pink. The values are elevated in the west, so we can so pink showed the same pattern as sockeye, indicating higher productivity. But something else that you can see is that they're much closer together in the West, indicating that they have more similar diets and that the diets were more divergent in the East. And this is what we've seen in the diets in our meta-analysis of diets for the Pan-Pacific region, is that in the West, the diets are more similar than in the East. All right, next slide. All right, so our next steps are just to, we're going to get into our fatty acid analysis this year, pending, further COVID shutdowns. We're going to integrate the isotope fatty acid and stomach content analysis to develop food-web interaction matrix and build our database of organism fatty acid profiles, so this nutritional value of prey. And it would be good to, bring in the other aspects of prey quality so we can do this very comprehensively, for instance, energy density. And then relate this nutritional health of salmon to prey quality. And then big thing is planning for 2022 with the aim to complete this Pan-Pacific collections using these approaches. And you can go to the next slide, and I'm happy to take some questions if there are any.
Sorry, Brian, I was thanking you and saying that you had answered some of the questions presented yesterday, but I was on mute. I think we'd learned that one. Okay, Mark, over to you.
Yeah, thanks, Brian. Dave Meerberg, in the North Atlantic, they have been finding decreasing energy density and size in some prey species like capelin over recent years. Anything similar in prey energy density in the Pacific?
Yeah, there was a study that came out from Alaska showing, I mean, Todd may even have been involved in that, showing decline in energy density over this period of heat waves that we had from 2015. So yeah, a decrease in prey quality within a species, not just related to a species shift.
Yeah, that was a staggering paper. 86% reduction in lipid energy content in the 1 year class. So yeah, let's hope it's not that extreme. Okay, one more before break.
Yeah, Dick Beamish asking, do you think there was any evidence that prey were limiting salmon growth?
I think that we might have a good indication or a further indication of this from the fatty acid data. One of the things that I do think is potentially an interesting avenue is the how much of this coastal signature we detect in the offshore environments. And I think the question I have is whether, like we saw in that it can be quite high. So they can those 1 year old fish can have a lot of coastal sort of biomass still in their body. Is that saying that there was really good growth in the coast, or is it saying that there was really not good growth in the open ocean? And I think that's one of the things we need to figure out. And coming back to this idea of first winter at sea, what's most important? Is it like this really, massive growth in the summer before? Is it enough resources to continue going through the winter?
Great.
Okay, Mark, we are at break time again until we get to, where are we going, 7 o'clock. And last couple of presentations and then our panel. So on break until 7 o'clock. Hi again. Welcome back. That we can do a very short introduction this time because this is my slot. And what we're going to talk about is Salmon returns to BC, Alaska in relation to the expedition. I'm going to comment on a few next steps. I want to comment right away, though, that the development of the salmon trends and the indices were totally dependent on many people, particularly Catherine Howard and Andrew Monroe with ADF&G. They provided a fascinating set of data that we'll be referring to. Okay, let's go on to the next slide. I think Chris Neville summed up our situation nicely in the sense that these are very new efforts and have little for really knowing what confidence we can assign to our observations at this point in time. I just wanted to remind people of the catch distribution to start. Just a couple of points here. Evgeny talk off about having the same survey coverage, and that's the region that I have noted here as no samples. He is, of course, correct. The survey design was very much more a replication of 2019 and 2020. Unfortunately, after we dropped some people off in Prince Rupert and came back out, we ran into a very large weather front, and vessel had to head south. So that's the primary reason for the difference in the distribution. I also want to point out that you will see different in the Chinook catches between the two surveys, but the large circle, the 21 to 50 size over by Vancouver Island in 2020, that was when the vessel had come back onto the shelf and caught, I think it was 25 Chinook in one set. So I don't really consider that to be applicable to the open ocean samples that we saw of 3 fish in 2019. Let's do a quick summary in the next slide, please, on terms of the catch of salmon. And this, of course, I noticed Alexi has changed a number from 2019, but these are the ones that I had available, and they're very, very similar. So one of the things that's interesting to us is that the number of trawls with 0 catches in 2019 was only 10 times out of 58. Unfortunately, in 2020, we had 23 of 52 trawl sets, so many more examples of 0 catch. But if we look at the species composition, it's different. Fortunately, we did catch a few more pinks in 2020, but certainly still not to what would have been expected ahead of time. And we did again see a more coal than I think we would have said that we expected before the trips. So those are points that we've already talked about. The asterisk on the sum for 2020 down below is, and this gets to the point of sets 4 and 5, I think, in the 2020 cruise, where we had about 70% of the fish caught in just 2 sets in 2020. So 2020 really has been observed as being highly heterogeneous in the distribution of the catches in the trawl sets. Well, let's go to the next slide. So a few overall comments that we've been making about the cruises. I mean, clearly overall the catch numbers were less than I say expected, but I note that we really didn't have a great basis for that. We think we all agree that we caught many fewer pinks than we should have seen compared to the relative abundance of pinks in commercial catches. The trawl catches were highly heterogeneous, and the species compositions were similar. The species were caught in different areas. We've talked about that quite a bit. But one thing that is interesting is when we look at the biological sampling of our catches, I say that they seem to be appropriate. They're surprisingly consistent with other studies where we might look at weight-length relationships. For Trump salmon, the weight-length relationship across the full range had a correlation of about 0.98. I mean, it was incredibly tight. And the other species, for limited numbers of samples, were very consistent as well. So let's go on to the next slide. And this is where I want to start thinking about, okay, well, what can we really learn from this, and how can we test what we've been thinking about? So the trawl net, somebody's talking in the background, please. Okay, the trawl nets that caught salmon for sure, but the question in our minds is, did we get a representative sample of the species and the relative abundance? And we have noted in a few talks that there really weren't any salmon predators caught, and certainly very, very few competitors. The abundance estimates that we've generated, Alexi has generated, They apply catchability factors based on extensive experience in Russia. But the question has to be asked is, are they appropriate for the surveys in the Gulf of Alaska? So there's an uncertainty there. The two expeditions were certainly informative, and we've heard many, many examples of that over the past 3 days. But, really, what are we going to be able to use in future designs? And this is where we get to what can we learn for the 2022 trips. So the way we might validate things is to test alternative gears, replicate sampling of vessels, alternative survey designs, use prediction based on our observations to date, and to create and test hypotheses for a number of the things that we've been talking about. So these are all things that we hopefully can come back to and discuss. We should note that the environments that we sampled in those years were seen to be unusual compared to past with the marine heat waves, but they were certainly surprisingly similar between surveys once we go back and do the direct comparisons. Let's go to the next slide, please. A bit more text here, but this is getting to what the talk here is mostly about. So based on our limited experience in these trawls out in the deep water, we were thinking that we would catch more fish, right? So the interesting thing is that our catches were pretty consistent or coincident with very poor commercial fishing along most of the Northeast Pacific coast in 2019 and 2020. The poor catches are very consistent with the surprising drop in the total North Pacific commercial catch in 2020, and I'll show you that in 1 second here. And in the Pacific Northwest region, many of the runs were very poor, and our fisheries were severely curtailed from many areas in Southeast Alaska, south through BC, and into the United States. So to assess the trend in the abundance of salmon through time, we've been compiling a set of data of our best indices of salmon returns over a fairly long period of time, but they had to include consistent estimates in 2019 and 2020. The state of Alaska has provided me 83 such indices, and I currently have about 110 in my possession, and I think we'll end up with about 150. So what are we using those data sets for? So what I'm doing is looking at the deviations over time from the long-term average value, and the long-term average requires a set of data from 1998 through 2018, estimate the average and look at the deviation through the whole time period and in 2019 and 2020. So let me go through now a few examples. I certainly am not going to do the 100 and plus of them. So the next slide, please. Now this is the total North Pacific catch. If you click the next step, please, that should be just coming there. This is a drop in 1 year across the North Pacific catches of almost 40% in a single year with fisheries throughout, and it's largely attributed to changing catch of Russian chum and Alaska pink and chum. So that is extraordinary. That's the single biggest change across the North Pacific we've seen over this entire time frame, and that's since 1970. Go to the next one, please. In Canada, probably, and we've heard the same sort of comment, the most famous population of salmon for us is the Fraser River sockeye salmon, and returns in 2019 and 2020 were certainly the smallest in history. Chris Neville showed you that. What I'm showing here is the last 22 years, but showing you the deviations over time within the cycle lines that Chris talked about. And you see that the 2019 and 2020 were certainly the most extreme deviations, but they are not unique in the trend or the direction. We have had a declining trend in productivity for several years. Go to the next one, please. All right, just click again, please. Now, these are three other BC, well, for us, prominent BC sockeye populations. And the only point I want to make here is these goes from the north to the west coast of Vancouver Island, and then we go down to the Fraser. And the same pattern actually exists. You don't see a unique major change in 2019 and 20. You see recent decline in productivity, pretty widespread for sockeye salmon in Canada. Go to the next one, please. This is a wonderful set of data from the state of Alaska. These are all of the sockeye populations where they have enumeration programs going on, 21 of them here. And these are the ones that all have rivers running into the Pacific ecosystems. They're grouped by region, so there's going to be 5 bars or deviations for each year. And you don't have to look at the detail, just look at the broad pattern. And then I've provided an overall average here so you can see the trends across all these populations. In the years since 2017, there would look to be a pretty strong reduction in productivity, but you've got quite a unique change for a few populations actually in 2019. So there's no simple story. Now, the other really interesting part, go to the next slide, there is an additional 13 indices in the Bering Sea, and this is Bristol Bay, the North Shore of the Aleutian, and up into Norton Sound. And all I'm showing you here is the dashed red line with yellow dot. This is the trend in the Bering Sea populations. And you can see you have a very different pattern in the recent years where the productivity and returns in the Bristol Bay has stayed very strong. Go to the next one. Okay, we're just going to change a few summaries here. So the top slide is the Alaskan chum, and all I've differentiated here is, again, the chum populations going into the Pacific and the chum populations going into the Bering. You can see that there's some coherence in recent years, but very definitely a strong message of a major reduction in 2020. The southern one is just a plot for pink salmon in Southeast Alaska. That is a major area of production for Alaskan pink salmon, and they differ in their even and their odd year abundance. So I've done the deviations within. It obviously doesn't matter. You see evidence of a strong reduction in production in the last few years. Go to the next slide, please. This is coho salmon. Coho, I've spent quite a bit of time looking at because, as Dick pointed out, this has been one of the interesting observations out in the winter surveys. These are 4 coho index streams monitored for a long time by the ADF&G in Southeast Alaska. So this is just north of the Canadian border, and I'll show you a northern Canadian population. Pretty strong evidence that there has been a decline in the production in Southeast Alaska for the last few years, going back to about 2015. So then if you go across the border in the next slide into the Skeena River, this is probably our most closely monitored Skeena coho population. It's called Toboggan Creek. It's about the central area of the river. And it starts a trend, which is interesting. So if you look at the deviation, there's a strong negative in 2018 and 19. But if there's any trend, you think that in recent years, it's actually a positive trend, very little deviation in 2020. This actually gets to be a much stronger signal as you go further south. So go to the next slide, please. Instead of the populations in southern BC coho getting to be poor, they're actually showing trends towards being a little better in the recent years. I've just chosen to show you what's a very problematic group of fish for Canada. This is a group of 11 different populations in the interior of Fraser. So this is above the canyon. And it limits many, many fisheries in southern BC because it's been in poor state for a long time. But instead of showing a decline in the recent 2 years, they're showing a positive response. So again, from the Skeena southward into Puget Sound area, probably, you're seeing probably an increasing trend in production, not declining. Next population, or next slide, please. All right, now this is a fascinating plot. This is a very different pattern. And many of you that in salmon will have seen other variations on this plot. But this is a group of Chinook salmon. There are 15 indices in Alaska that they've monitored for a long period of time. The blue bars are the Alaskan index, and you don't see much variation here other than a huge change in the mid-2000s, 2007. Now, the red one is actually the Canadian border Chinook, and this is so well up the Yukon River as it goes into Canada, and you see it's doing essentially the same thing. I'm not sure what it was doing in 2000, but that's certainly an outlier there. I go on from here, please. Next slide. Now, if you try to actually do a simple summary like this for Chinook salmon south of Alaska, it becomes much more complicated. And there are a lot of people that monitor this. And part of this is that you get into very different survival patterns with different life histories of Chinook. So I'm not going to spend a lot of time on this. I included a figure from Tom Quinn's book because it very nicely shows the currents going up the Northeast Pacific coast. And there's a lot of data from Côte tagging and genetic sampling that shows that many fall Chinook and summer Chinook stay on the coast and migrate up the coast into Southeast Alaska, Central Alaska, and then even into Bering Sea. But what we're seeing recently for many summer and fall Chinook is that they're showing better production cycles than they were a few years ago. There's no question that spring Chinook yearlings are the ones that are doing most poorly in the southern area. And go to the next slide, please. I included this one on chum because we do have the stock ID. I'm not going to spend a lot of time on it because Iwawa-san did such a nice job. I have tried to compare the age structure in our samples to the northern and central BC chum returns. 2019 doesn't bear a close resemblance, but 2020 is very similar to the age structure of the fish returning in central BC. And I think it's because of the figure above where almost 60% of the fish that we sampled were from BC. So it's quite possible that in 2020 we did sample a section from that area. Okay, I've got a few summary slides to finish up here, and this is more to, next slide please, summarizing a lot of information and then looking forward. So my summary of the salmon is that sockeye over a wide area have done poorly in the recent years, but it's not specific to 2019-20. It is a consistent trend over recent years. But Bristol Bay, Bering Sea populations, they continue to do their own thing, and they have sustained production. Pink and chum production has been very poor in recent years in Alaska. Chum continues to be poor to very poor through north and central BC, but pink salmon are much more variable. There is some decrease, but mostly it's average or better, right? The really strange exception is Portland Inlet and the Nass River, where we see actually a pretty good return of pink and chum to that one river. Coho abundance has been very poor in the north, but as I said, it improves as you go southward, and I really don't have much data from the southern US yet. I'll receive that shortly, I hope. And we talked about the Chinook already. Fascinating trend in Alaska in that index and the change in about 2007, but it's very difficult to summarize all the others, except to say that for summer and fall Chinook in BC and into the southern states tend to be more positive than negative. Okay, the next slide, please. As I've talked to a number of people, and I made a comment about this yesterday, I think as we go forward, there's more than just the abundance that we should be considering. The abundance trends we will have in a database, so that will have, well, it's over 100 indices now. I'm thinking, well, 140, 150. Many people have also documented changes in the biological characteristics, particularly size at maturity, age at maturity, and returns. So I think that a database of that would be useful and is actually something that's very doable. Dr. Beecham gave a nice talk about the state of the stock ID. We really need to finish that up so we can actually do some of these assessments when we're out there sampling. And I really think that run timing is something that we need to really consider. When I was talking with the ADF&G people, I was surprised to hear that 2020 Bristol Bay was one of the record returns. I had heard that it was very poor. It turned out that the run to Bristol Bay was a bit late. And as you go down the coast, you get the same comment. Late returns in chum, some early returns in pink. I think this is something that we should be picking up on. And a major point, I think, has to be that the data quality over a large area is so variable and so inconsistent in types of indices that if we're doing studies together in the future, I think we need to define a set of indicator streams that we all maintain. And it's a really good way to involve our local communities in being engaged in the studies. And go to the last slide. So next steps, I just want to say that Dr. Beamish and I were recently funded to do the GillNet study with the March 2022 trawl surveys. So we will be able to do some parallel fishing and looking for different species composition. We were also funded for 2 researchers to work for the next year and a half to really determine what would a full scientific study be to link climate to oceanography to salmon production, well, fishery production in total. That can start fairly soon. And we are actively working with Mark and MPSC in terms of how you develop a large multi-year international program to really link the climate change to oceanographic features to fish production in the North Pacific. Some of this is likely to be picked up under the UN Decade of Ocean Science, but hasn't been at this point. And we all agree that one of the things we need to do is get rid of gray hair, Beamish and Riddle, and find younger scientists and to build communicators. And my final slide is just, go to the next one, please. Is really just a thank you to all the people. I'm not the bearer of these indices anymore, so I have to rely on many other people to provide the data sets so that I can analyze them. And I freely share the material from Lori, even though she doesn't know it. So thank you very much. So can I ask myself a question?
Go for it.
No.
Go easy on yourself, though.
I will. Hopefully we can pick up on the next steps and where we're going together in the discussion, and that particularly leading from the March 2022 cruise, which is largely designed with science team, but from there after, if we're casting models, etc.
Right. I'm not seeing any questions yet, Brian.
What time does Jim start? 9:25, 2 or 3 minutes. All right, so. Well, if there's no questions, then if Jim's available, why don't we just go ahead?
Sure.
And that, so oh boy, is Jim available? And are we ready to go with the next presentation? Okay. If there's no question, then let's just go right on.
There we are. Okay, well, thank you, Brian. Thank you for folks inviting me to come talk about some of our coastal surveys. These are, I'll be talking about 2 surveys that we conduct here in Alaska, and I specifically am going to talk about the forecast models that those surveys support. And there's a number of different folks that are involved with the surveys, and they're operated as both the surveys and the forecasts are cooperative efforts by the Alaska Department of Fish and Game and World Fisheries. And I just mentioned a few folks here as authors on the presentation. There have Sabrina Garcia, Andy Piston, and Steve Heinel, and Sarah Miller, Rich Brenner, and Zach Liller are all with Fish and Game, and of course myself, Jamal Moss, Wes Strasburger, Emily Ferguson, Andy Gray, and Ed Farley. We all work at the Alaska Fisheries Science Center, which is also known as NOAA Fisheries. So the two surveys that we still maintain, have been maintained for several years now, are the Northern Bering Sea Surface Trawl and Ecosystem Survey and the Southeast Alaska Coastal Monitoring Survey. The Northern Bering Sea Survey started about 20 years ago, and this was part of the NPFC Basis Research Program that started in 2002. And currently it's supported by the chartered fishing vessel, the Northwest Explorer. And this survey provides forecasts or harvest guidance for Yukon River Chinook salmon fisheries. The other survey is the Southeast Alaska Coastal Monitoring, or the SECM survey, that started about 25 years ago and is supported by the Alaska Department of Fish and Game research vessel, Medea. And this survey, one of the main forecasts it supports is the pink salmon harvest forecast in Southeast Alaska. Sorry, we'll go on to the next slide. I'm sorry. And yeah, these are the two vessels that support the surveys, and we'll go on to the next slide too. Thank you. So this is the survey grid for the Southeast Coastal Monitoring Survey. This survey occurs within the northern region of Southeast Alaska. So in the upper right-hand corner of the figure, you can see that area in the panhandle of Alaska. And so it samples there in the northern region. So there's 8 stations within icy straits that have formed the main monitoring stations. And they're sampled monthly between May and August. And during May, the sampling is just for oceanography, but June, July, and August has included both surface trawl and oceanography. Actually, for this year, 2021, the August surveys will be discontinued, and we're going to try to expand the July sampling to get a little broader coverage of Southeast Alaska. And the survey includes surface trawl sampling, of course, for juvenile salmon and other nekton species. It also includes bongo net sampling for zooplankton and CTD operations. So next slide. The work, I kind of group it into 2 related categories, including juvenile assessment and juvenile ecology. Of course, the surface trawl catch rates, we use that to describe the distribution and abundance of juvenile salmon and other species. We collect information on codawire tags. This would be Chinook and Coho for origin. We collect information on thermal marks for sockeye and chum hatchery thermal marks. And we started doing some work on microsatellite genetic stock identification of juvenile Chinook just recently. We collect a number of different aspects of the juvenile habitat, but the one environmental variable that is used in the assessments is the icy strait temperature index. The survey also supports a wide variety of other objectives, monitoring and studying aspects of the diet and feeding ecology of juvenile salmon, as well as monitoring changes in their size, growth, and condition, and just overall ecosystem conditions within Southeast Alaska. Go on to the next slide. This figure shows the trawl catch rates. So this shows the distribution of juvenile salmon in the different habitats and over the different months that the SECM survey has occurred. And these tan bars are the strait habitats. So you can see that the juvenile salmon species, all of them except for Chinook salmon, reach their highest densities in the strait habitat during June and July. And that's the main reason why the strait habitat has formed the core monitoring component to the survey. And now Chinook, they tend to remain in the inshore or nearshore areas, and that's through the whole season. And we added stations in 2018 to help address Chinook in the inshore stations. So we'll go on to the next slide. The juvenile abundance index for the pink salmon harvest forecast models are based on the peak catch rate, and that's either in June or July. And we use vessel-specific fishing power coefficients to standardize the catch over time. We also include, as I mentioned earlier, we include the isostrait temperature index, which is just the average temperature in the upper 20 meters of the water column at the stations in isostrait. Forecast model is very simple, just a multiple linear regression model. And we assume a log normal error. And we use an 80% bootstrap confidence interval to define the range. Go on to the next slide. These three figures here, and you've seen this, folks have probably seen this before, this kind of captures the components of the forecast model. The figure on the left is the model fit, so this is our predicted harvest versus the observed harvest. The figure in the center is the partial residuals for catch rates, so this is, you can see the effect as our catch rates of juveniles increase, we forecast a larger harvest. And then the figure on the right, which is interesting, is the relationship with temperature. And so there's a number of different ways in which we can interpret temperature. I think what might be occurring is, since we're only sampling the northernmost corridor, but we're forecasting harvest to the entire region, I think there may be a temperature effect in terms of our trawl catch rates as it applies to juvenile abundance. So basically, in warmer years, I think there may be more juveniles that exit Southeast Alaska or migrate through the northern corridor more years. And so there's that temperature correction in terms of how our trawl catch rates are related to juvenile abundance. So we'll go on to the next slide. This is another way of just showing the model performance and fit to the harvest data. The solid lines are the model fit, and the bars are the actual harvest. And then you can see that the forecast for 2021, which is just above 20 million fish, we're forecasting an increase in the number of pink salmon harvested in Southeast Alaska for 2021. So next slide. The other survey I want to talk about is the Northern Bering Sea survey. Again, it started in 2002, but we used a different trawl design, a much smaller trawl design. So I tend to use data from 2003 on. The survey occurs during the month of September, so that the juveniles have actually been in the ocean for about 2 to 3 months by the time we sample them. And again, the survey includes surface trawl sampling for salmon and other pelagic nekton. It includes zooplankton sampling with bongo nets and CTD data. And this year we're going to add beam trawls and benthographs to our sampling activity. Next slide. So again, grouping the salmon research into two related objectives. One is juvenile assessment, the other is juvenile ecology. The assessments are very important because to the survey, because they provide funding support for funding sources like Alaska Sustainable Salmon Funds, and they provide support for estimating juvenile abundance. We also collect information on habitat, like mixed layer depth, and that's used to help correct for vertical distribution of salmon. So that is part of the juvenile salmon research, and this is in particular part of it's an important part of the juvenile Chinook salmon abundance estimates. And we generate stock-specific juvenile abundance estimates when we can, and so currently that's for juvenile Chinook and juvenile chum salmon. And we also conduct research on juvenile ecology. It's important as it provides an ecosystem framework for the juvenile assessments. And the key focus of the juvenile ecology research is really tied to the impact of warming climate in the North Bering Sea and the loss of Arctic sea ice. And it's really through the understanding of juvenile ecology that, I think we can begin to hopefully define what to expect as the Northern Bering Sea warms and as we lose Arctic sea ice. Go on to the next slide. This figure shows the distribution of Chinook in the Northern Bering Survey, and the symbols on the map are the spatial center of their distribution. And as you can see, they're just to the west or southwest of the Yukon River Delta. That's where the Yukon braids out into the multiple distributaries before it flows into the Bering Sea. And this is mainly because juvenile Chinook have historically, juvenile Chinook from the Yukon River have been the most abundant stock group in the North Bering Sea. And we are starting to see some changes in the mixtures of juvenile Chinook salmon with the recent warming of the Northern Bering Sea. Next slide. And so these four figures show the change in stock mixtures in North Bering Sea. In particular, I want to point out the figure on the lower right. You can see that's for the Upper Yukon or Canadian origin stock group, and it's been relatively stable, around 46%. But in the last couple of years, we've seen a fairly significant decline in the proportion of the Upper Yukon stock group. And then the figure on the lower left is the proportion of other Western Alaska or non-Yukon stocks. And so that has started to increase significantly. And I think this is indicating that there's a couple different things. There could be a couple different things going on, but there is definitely movement, I believe, from Chinook salmon from the Southern Bering Sea into stocks rivers like Kuskokwim and Nushagak. And so that's altering the mixtures and altering juvenile migrations in the Northern Bering Sea. Next slide. So by taking our trawl, catch, and abundance estimates and combining it with stock mixtures, we can generate these stock-specific juvenile Chinook salmon abundance indices. And as you can see, they follow a similar pattern over time, and that's just mainly because the Canadian origin stock group has remained relatively stable. And I should add that the top figure is for the Upper Yukon stock group, and the bottom is for the total Yukon River Chinook salmon abundance. We see a bit of a steeper decline for the Upper Yukon stock group, and that's mainly because of that change that we're seeing in their stock composition. Next slide. So if we take juvenile abundance and spawner abundance, we can capture how the early life history survival of Chinook salmon is changing over time. And so the dashed lines on these two figures are the spawner abundance, and the bars are the number of juveniles per spawner. And again, the top figures for the Canadian stock group, and the bottom figures for the total Yukon. And as you can see, both figures highlight a significant decline in this early life history survival of salmon. It's particularly important to note that some of the recent years of low abundance have come from relatively good escapements, and that's what stresses the importance of this decline in survival during the early life history stages. And I will say I'm kind of using survival very loosely here, because juveniles per spawner can be affected by sex ratios, fecundity, and possibly even pre-spawn mortality in Chinook salmon. Next slide. So the abundance of Chinook salmon is important because we've been able to establish that juvenile abundance provides a reasonably good indication of what to expect in the future. So we use these very simple models of juvenile and adult abundance to project what future returns will be for the Yukon River. And these models indicate that much of that year-to-year variation that we see in returns is occurring during these early life history stages. Now, it doesn't mean that survival after this stage is not important. It just means that has been relatively more stable than some of this early life history survival. The variation in these models is important. We use the 80% prediction interval to define the range of forecast, range of run sizes that would be returning to the Yukon River. So next slide. So it's combining them all together, the models I just showed in the previous slide, juvenile abundance, and we also use the average 3-year maturity schedule. We can create these projections or these outlooks for the returns to the Yukon River. And so both models indicate a declining run size for the Yukon River through 2022. And so that basically captures the context or our projection for what to expect in the Yukon River for the next couple of years. We'll go on to the next slide. Now, the way we take the juvenile abundance and we combine the juvenile forecast and we combine that with the sibling models and production models, so the Ricker model here. So you can see the 3 models here on the left. And actually, Dr. Curry Cunningham was instrumental in helping us revamp the entire sibling model this last year. And so it's all dynamic models, dynamic time-varying coefficients, and and it's all done within a Bayesian framework. So for the first time, we're starting to see actually similar performance with the sibling model and the juvenile model. So that's giving us a reasonably good confidence in what to expect for this coming year. The figure on the right is the combined forecast for all three models. So this is an inverse variance weighted probability distribution for run size. And the lines on the figure on the right are the confidence intervals. So the solid line is the 80% confidence interval, and that's what we are using. That's ultimately what comes out to be the guidance for this year, which is a run size between 42 to 77,000 Chinook salmon, Canadian origin Chinook salmon. So next slide. So I'm going to just an overview. The coastal salmon surveys in Alaska. There are two of them that provide harvest guidance in Alaska, and one is for Chinook salmon fisheries in the Yukon River and pink salmon fisheries in Southeast Alaska. Now, we do have some forecast models that are developed for other species, but they've not yet been integrated into an official forecast or harvest guidance. The coastal surveys were originally established by Alaska Fisheries Science Center, or NOAA Fisheries, but these are now operated and run as cooperative surveys between Fish and Game and NOAA. And this is largely due to their current application to salmon harvest guidance and to run size forecast. And these forecast models have been just instrumental in securing that annual funding support that has been required to keep these surveys going. The two surveys, one's been running annually for 20 years and the other one's been running annually for 25 years. So some of the key characteristics of both of these models are that they're based on juvenile abundance estimates. These are trawl-based juvenile abundance estimates. And they also include information on juvenile habitat, and in particular some of the C2D data that we incorporate into both of these models. And it's really, we use that environmental data to account for changes in the distribution of juveniles, and as well as just correcting for catchability of the survey over time. Both of these models are fairly extensively reviewed by both state and federal fisheries biologists before they've been used or before they were used in harvest guidance. And then finally, and I think this is kind of important, we integrate information on juvenile ecology, kind of feeding, diet, growth, and condition. So we try to always add that as sort of an ecosystem consideration to the forecast or the outlook. And I think this really helps to kind of set a tone or enable these precautionary approaches to harvest guidance. So that is all that I have. I'll be happy to take questions.
Thanks, Jim. Well, Mark is picking up on questions. I've got one for you. So we're talking about recent years, and that I'm interested in whether the Southeast Alaska forecast model, which, as you described, is a simple multiple linear regression, and that, are you seeing increased deviations from forecast in the recent 2 or 3 years, or has it been very consistent with past?
Yeah, no, we're not seeing an increased deviation. One thing that we do see when you look at the performance of the model over time is that actually the model that has been used to forecast harvest over time has changed. So what I showed you is the fit of the current model. And but if you look at the actual performance, it's not as good as the fit of the current model. So there's a couple of things going on there. But the current model that we have seems to be working, and it actually fits better than some of the earlier models or earlier versions of it. So there's a lot of different ways in which we can, like we're applying slightly different vessel calibration coefficients, or we're using slightly different weighting in terms of how samples are collected each month. So there's just subtle changes to the model. I would say no, it's actually working fairly surprisingly well.
Okay, Mark, questions?
I'm not seeing any, Brian.
Well, let me go on.
Be quiet. Okay.
Yes.
So the subtle changes you've made, are these new environment parameters, or are they changes in how you estimate catchability, or what is a subtle change?
Well, the fishing power coefficients, we've had like species specific fishing power coefficients. So like chums and pinks and GOA. There were some fishing power experiments that were conducted. And so there's some slight differences between the species. And so one of the changes we just did recently was as we decided to just go with a single coefficient for salmon, apply it's simpler and in terms of because we've got multiple vessels and multiple fish species. There are many coefficients to try to keep track of, and I think that's where actually simplifying it actually made the models fit a little better. So and then you have to go back probably 5-6 years ago where there were like ecosystem ranks. There were other ways of approaching environmental covariates, and there were some cases there could be 3 or 4 environmental variables, and so we've kind of just narrowed it down to just pick 2 that work well. Because you start to begin to overfit the data to the error that's in in there, and it may not really be a stable. So trying to keep something that's fairly stable, use the same model from year to year, so that we don't we can begin to see how performance characteristics of a given model. It's a long way of giving you some insight into the subtle changes.
Yeah, thank you.
So we do have a question here, Brian. Wasn't signed, so it's anonymous. Thanks, Jim. Can you share any insights into the juvenile salmon ecology side of the monitoring programs? Have you seen any changes in some of those ecological parameters, example, diet, habitat use, etc.?
Oh yeah, good question. So we it really depends on what survey you're talking about. And for Southeast Coastal Monitoring, one of the key things that we try to include with every forecast is conditions in the Gulf, kind of the marine heat wave. Is it a warm year, or what's kind of generally happening there? We haven't really been able to connect a clear linkage to our forecast or survival, but it's that's been something we've always included in the forecast. We also keep summaries of like juvenile size to say how well they're growing, are they in good condition. So those would be just kind of considerations that would be added to a model. So we did see size and abundance decrease pretty clearly back in like 2017. So we've started to see growth rates increase a bit and abundance increase. So that's good. So we're kind of seeing, in addition to seeing higher catch rates, we're also seeing better growth rates of the juveniles in Southeast. In the North Bering, one of the key things that we see is that we're seeing declining feeding stomach fullness and feeding intensity. So clearly there's a declining, we think it's prey availability, that we're just seeing less food available for juvenile salmon. So the amount of food in their stomach is declining. It's declining with temperature. So that's just a concern. We haven't been able to link it specifically to a forecast model, but it is something that we bring up and we discuss those concerns when presenting the forecast.
Great, thanks. Question from Michelle Crown. For species with complex age structure, how do you account for age at maturity and size at maturity in your model?
Yeah, so that's really interesting because, we always think like, oh, it's survival. We have to be really careful about how survival affects our juvenile forecast models. And that's true, and it's very obvious that changes in survival during their high seas life history stage will alter that model. But changes in maturity schedules can also affect how juvenile abundance affects adult returns. And this is particularly true when you have adjacent years a very different brood year strength. So when juvenile abundance is very different from one year to the next, it becomes very sensitive to having correct estimates of maturation. So how we just take the last 3-year average, and we just assume that that's what the maturity schedule is. But I know that introduces error in the forecast. And one of the things that we hope to do is to be able to include things like the size of the juveniles. That's one thing that we see when juveniles are growing well for Chinook and North Bering. They're I should say, let me explain. The size of the juveniles is highly correlated to the jacking rate. So when the juveniles are very large, there's a higher percentage of them that return early. And so that's where I'm hoping that maybe we can take information on the ecosystem and juvenile size and be able to predict maturity. If we can predict maturity, then that's going to be something that will improve our forecast quite a bit.
Okay, Mark, we are past our time limit, so thank you very much, Jim. And I think I hand this over to you, Mark, for the panel discussion.
I think that's true. Yes. Oh,
there is a break. Okay. Sorry, I did not have that on my schedule. So what time are we starting? 8 o'clock is the start time. 8 o'clock. Okay. Thank you, everybody.
okay. Thanks. Hi, everybody. Mark Saunders here. I've got the pleasure of hosting this panel for the remainder of the evening and bring you a little more information, more about where we go from here. So I'm going to start off with a bit of a presentation on the 2022 expedition, and follow that up with Dick Beamish and Brian Riddle giving you short overview of Gillnet's survey. They are planning to accompany that 2022 trawl expedition. And then we'll move into our work with a panel. And we've got 7 participants that are involved very heavily in the planning of the 2022 expedition. So we're going to challenge that panel to help pull out what we learned over the last 3 days and how those what we've learned, those lessons learned or findings or contributions are could impact how we plan the 2022 expedition. So we'll try to pull that out of them, and we'll look to the audience as well with the Slido to participate in bringing some of those questions and observations forward to the panel. So I'll start by giving you a little overview of the 2022 expedition that we are planning. This follows on the work that has been developed by Brian, Dick, and a host of partners, and articulated through the International Year of the Salmon and the North Pacific Anadromous Fish Commission. The idea came from Dick to make sure we need to get back into the high seas, and Russia was very prominent in bringing forward this idea of a Pan-Pacific expedition, a fully ecosystem survey that would cover 5 zones that you can see there, and a number of our talks over the last 3 days have highlighted some of the previous work that's been done out there, but getting back out there with a high degree of higher amount of coverage than has been seen for many decades, and have the capacity to start to utilize modern systems and technology like genetic stock ID, etc. The status right now, we've applied for, we have a number of applications sitting in with various agencies and countries, and we have notional indication from Russia that 1 ship will be provided for the Zone 1. Zone 4 and 5 would be covered by a Canadian vessel, the new ship, the Franklin, and American ship, the Shimada, but all 3 of those vessels are notional. We're still waiting formal approval through ship scheduling processes in each of those countries, and we are raising money for zones 2 and 3. We require about $3 million for charter. We have a Russian vessel that is available to utilize that funding in charter, and we've raised about $800,000 of that with a contribution from Canada, as well as the North Pacific Research Board has provided some funding for that. So we continue to work on securing that. The overall objective is to demonstrate the utility of an international Pan-Pacific winter ecosystem survey, to understand how increasingly extreme climate variability in the North Pacific Ocean and associated changes in the physical environment influence the abundance, distribution, migration, growth, fitness, and survival of Pacific salmon and surrounding species. Key here is, as we've been working over the last 3 days, is really working towards understanding the mechanisms and how it can be related and inform management systems. I won't go into this in detail, but you can have a look. Very similar to what was done in the Gulf of Alaska expeditions. These are the proposed sampling protocols that we're working across the 5 countries to agree on these protocols, and they're continuing to evolve as we plan. I'd like to give you just some of the folks you've heard talk over the last couple of days that were involved in 2019 and 2020 from the Canadian side are also involved with some funding that we got through the BC Salmon Restoration and Innovation Fund. We got about $3 million to support Canadian academic projects, and I won't go into them in detail, but you'll recognize some of the names. And we're working in areas around the physical oceanography, chemical, biological oceanography, related to some of the technology and projects that you heard about over the last couple of days. Another suite of them here, acoustics. I'm particularly excited about this. We didn't hear much about the acoustics, but I think it's a tool that's going to be very important in terms of linking and building our understanding of the ecology. Yeah, right through the genomics, work on the distribution using otolith microchemistry, etc. One of the things we haven't talked about, although I think it was alluded to by Brian Riddle about the issue of data and making it available, certainly looking at it from the integration of our understanding with returns and trends over time, and this whole investigation and work that we're doing across multiple countries is our research is really challenged by our inability to mobilize data. We've been working with Tula Foundation, the Hakai Institute, in partnership, and to make the data develop open source approaches and standardized approaches that adhere to the FAIR data principles. And so we're working with the 19 and 20 data and our data management plan for 2022 that is looking very positive. It's also, these expeditions are an opportunity to test multiple emerging technologies, as you've seen some of the genomic work, but we're also planning, we've got several projects that will include autonomous vehicles, gliders. The one pictured is a collaboration between the University of Washington and University of Alaska Fairbanks, where we have a glider that's equipped with a hydroacoustics package and with some new technology to allow it to compress data and talk to satellites. We can potentially use these. We'll be putting these out in advance of the survey and in conjunction with the survey to augment and see how, going into the future, we can possibly use these to augment or replace expensive ship time. We also have a Canadian project, Jody Climac and others with the University of Victoria, that have a set of gliders that will be deployed as well. So there are some contacts there if you've got further interest in understanding, getting involved and working on, have questions about this. I want to point out the expedition countries that we've got. Canada, Jackie King will be on our panel. You may have heard some questions from her over the last couple of days. Japan, Urawa-san and Sato-san. From Korea, Sang Seong-yoon. Russia, Alexei Somov, and from the United States, Ed Farley and Lori Whitecamp. And our chief scientist is Evgeny Pahomov. So I think with that, I'm going to give you a sense of what we're up to, and now move into the panel discussion and try to get some, try to bring together what we've learned over the last 3 days and things that we learned and how could they inform how we go ahead with this really very bold, fully trans North Pacific set of surveys. So our panel includes folks that are very involved in the research to this point, but as well in planning. So we've got Dick Beamish, Brian Riddle, Jackie King, Alexei Somov, Lori Whitecamp, Evgeny Pahomov, Vladimir Rychenko. So to start moving through this, what I've got sort of one question for really that will fuel this this session, and I think it's the next slide that we've got. If you could put it up. So the question to the panelists is one major outcome of the 2019 and 2020 expeditions that stood out for you, and how could this outcome inform planning for the 2022 Pan-Pacific Winter Expedition? I know most of you will probably have a couple of them, and we can cycle through the group. But I'd like to hear your first thought about an outcome that could inform planning for 2022 expedition. So I was going to give the first shot to Dick, but I think I'm seeing his panel is dark, so I don't know if you're there, Dick. He's not here. Okay. Well, let's go to Lori. She's the next one I'm seeing on the panel. So Lori, thoughts?
You know, one of the things that really stands out to me, especially compared to previous surveys, is how valuable the genetic data is, that we can take individual salmon and be able to say, this came from here. And so like Urawa-san showed today with the chum salmon work, be able to put it together for salmon, chum salmon from different populations, where they go, et cetera. And I think it's really critical that we all use the same baselines as we're sampling across the Pacific and have this common body of where these fish came from. And especially in the case of like Chris Neville's talk, where she's able to look at Fraser River sockeye as they're spread all the way across the North Pacific. So using that information about salmon that we can only get from genetics to really build the foundation for our understanding of what salmon are experiencing, I think is really key.
Thanks, Lori. How about you, Brian?
Thanks, Mark. Actually, I would agree first off with Lori. I think it's invaluable to use these new tools. And so I would endorse that strongly. I think we need to recall the message from Terry that our weakest point is pink salmon, that we should be addressing directly, and it will be an international collaboration required to get the baseline. We can do it simultaneous with the cruise, so we can do it after the fact, but we need to do it. And then I think thing I'd add with the stock ID, it's the only time that we heard it was today and talking about the energy content, but I think we need to use the otolith marks to identify the hatchery wild component because we certainly still get many criticisms about the large-scale hatchery programs, and we need to have a better understanding of that. So stock ID, otolith microchemistry for the hatchery wild. If we're just doing one each, that's mine.
Okay. Thanks, Brian. Well, why don't we jump over to the other side of the North Pacific? Alexi, what stood out for you? I think you
must be muted. Yeah.
Sure. Okay, Jackie.
I thought I was just going to freeze while you worked out the mute problem. And maybe this is just, my interest as an ecologist, were sort of the number of questions that the two surveys actually raised. One of the most interesting, I think, is a lot of the diet stuff for salmon. I'm intrigued by the fact that not a lot of mactophids were a huge component of the salmon diet in the Gulf of Alaska, at least when we were out there. I like Brian Hunt's work in looking at, being able to tell us that, yeah, those squids and mactophids are actually competitors for salmon. So for me moving forward, I think, the two cruises are already firmly placed in ecology, and they've got a lot of new techniques applied to placing the salmon in the food web. So we need to continue those. Of course, what is most exciting and I think it's because 2020 had bad weather and the northern part was missed. It really does highlight the need for complete coverage or distributional coverage. I know there's been a lot of talk about being able to, sort of focus in on certain stocks, particularly if they're associated with fronts and eddies. And I think that's extremely useful and interesting, but we still have yet to fully cover the whole range of distribution. And I'd like to see just the basic ecology of what's going on of these fish in eastern versus western Gulf of Alaska, actually. So I just thought a lot of the diet stuff and the trophic level stuff was really great, and it opens up the question of how are they making it through these winters? Really, when you put it all together, they're phenomenal animals.
Great, thanks. So did Alexi, how did we make out there?
Does anybody hear me right now?
Yes, we do.
Wow, that's fine. So I just wanted to say that it is very significant that we have results of these two expeditions in 2019 and 2020, because one of the main tasks for the following expedition is to have a big picture of the salmon, of their distribution. Where are they in the North Pacific during the winter? And having results of two previous expeditions, we understood that we should cover more area. So I mean that negative result it's a result as well. So knowing that in the Gulf of Alaska we caught not as many fish as we expected, it means that we should broaden our study area, especially goes to southern region in terms if we want to understand pink salmon and coho salmon distribution. And moreover, I would like to emphasize that these two expeditions showed that ecological processes go in some kind of different ways between northwestern and northeastern Pacific, just as Jackie King said. So I agree with it, and so we cannot just directly compare what is going on in the northwestern Pacific and just to project it to the northeastern.
Thank you. So Dick, I'm glad to see you back. So your take on a major outcome and advice for 2022.
Somebody crashed my computer here.
As the
I had to
puppy.
get Ann to get it to work again. Anyhow, I poured myself a glass of wine while she was getting it set up. So here we go. I think Jackie King did it.
I snuck over there. You're right, Dick.
I almost called you to come down here and fix it. Just a few things. This is one of the few times that we've really had an international team of researchers focus on a large scale, or let's call it a big picture. We've all been to numerous conferences, and recognizing that this is virtual, that in itself is new. But this, what we have just done, and the two expeditions that included international teams, we are moving towards developing teams that will focus on looking at a bigger picture. So we're on our way if we can continue doing this. In terms of the next expedition, we'll see how it develops. I like the idea that Riddle and I were able to. I think we've got our money, and I like the idea of using some different gear. Both longline and gillnet, and I told Jackie that we're intending to follow her around, and We had a question that we didn't answer about catchability during the day and night. And my experience with trawls is that the nets catch better. The catchability is better during the day, but the Russians have more experience with that. Anyhow, my point is that I think it's really good that we will have other gear, including long lines and as well as the gill net. So I don't really have a lot of comments on how to design the 2022. I think the people that are putting that together have done a good job. But what is important, I think, is to follow up on some of the issues that we talked about. Now, at the beginning, I mentioned that the 2020 catches were back to the mid 1980s, and Brian Riddle showed that. And that's a huge issue. And that really hasn't happened in the careers of all of us that are on this call, and including our colleagues. So we are possibly at a time that's unprecedented in terms of what we need to know to understand what's going to happen in the future. And I hope that one of the things that we learned from the expeditions and from the conferences that the conference that we just had is that there's a huge part of the understanding of what regulates salmon abundance that we don't know. Now, I think the evidence is accumulating that the brood year strength is mostly determined at the end of the first ocean winter. Mortality occurs after that, but as we've heard, it tends to be constant. So, in summary, the concept of an international team of researchers trying to see a bigger picture, recognizing that it's the ocean that is regulating abundance after you get a reasonable number of juveniles entering the ocean. And we need to see that bigger picture. So we're on our way. We'll see what happens after 2022.
Mm-hmm. Thanks, Dick. Evgeny.
Can you hear me? Yeah, fine.
Yeah, yep. Go ahead.
Okay.
Yeah.
I think I'll maybe mention couple of things. First one is I was really impressed by the power of eDNA, and this was something which gives us a very, very valuable tool. But it also connected to the problems which we highlighted in some of our presentations, saying that we are not really capturing very well actual food prey of salmon. We are not capturing well FOZs, we are not capturing well some other. We do capture myctophids and squid, but it's not always the most important food item of the salmon. So, and I think that eDNA in combination with probably extra gears as well as acoustics would probably provide us with different layers of understanding other trophic levels rather than between small zooplankton and the fish. The second point was already brought by Jackie, and I think it's actually competitors question, and I think that was kind of eye-opening as well, because there is always inferences that big pink salmon create competition for other salmon. However, what we've seen in Chris' presentation and then in Brian's presentation, that pink and Sakai, for example, do not overlap that much. Actually, pink separated, so there is no spatial overlap, and as a consequence, probably very low competition. But what we do see, and it's much more complicated, we see more real competitors to salmon, such as squid, such as myctophids, and some other organisms, perhaps which we are not really capturing. And I think the the last thing, which is related to the food web work which we are doing, is that there are quite significant and subtle differences between East and West. And I think that's something which we need to pay attention to and continue working on. Yeah.
Who have I missed? Vladimir.
Vladimir, yeah.
Thank you, Mark. I will be a little simplify and generalize, but since I am last in this famous panel, maybe it would be suitable. So what is the most important what we have found? Actually, if before scientists consider high seas for salmon like a black box, they are going somewhere, they compete each other, they suppress each other, and something like that. Now we have found, and it was confirmed by these expeditions for the northeastern Pacific, that salmon are much smarter than we consider them. They don't overlap the areas as Chris Neville shows for sockeye and pink salmon. They can found these special biotopes. They are differ by diets, and it also means they are searching for food in different places or in different layers. So what we need in a future expedition, we need to more focus it on the biotope study. We need more oceanographers and not only technical staff as we had in our expedition 2019. And I'm not sure who did this oceanographic work in 2020. We need the people who will think about that biotopic picture of the ocean, or the winter ocean. And we all need go from common thinking to scientific thinking, considering the salmon distributions at high seas. You mentioned the hydroacoustic. Yes, I also think that it's areas that should be significantly improved. We have some records from 2019, but I am not sure whether somebody looked through it significantly or not. And we also need a specialist who can recognize this, maybe not a species, but just types of animals who will be reflected by hydroacoustic. For example, in 2019, we with Evgeny saw on the hydroacoustic device a big lump of something moving just a little bit deeper than the layer of our troll. And we caught several dozens of these sergestid shrimp. But if someone can reach this food resource, it would be great for them. It's caloric, it's a high, powerful resource, and usually someone easily and readily eat shrimp in the near coastal environment. But we don't know how many they are there in deeper ocean and how they behave in the daytime. So it's a big information that's still hidden from us. Currently, actually, figuratively speaking, we just scratched the surface and looking what we have under our nails, and we even more surprised. But if we will take a shovel and we'll try to dig, and maybe we will find much more information about how this ecosystem functioning and how the productivity of this winter upper layer is supported by deeper layers. And very likely that we will find predators and competitors in these deeper layers also. Since actually we in our expedition used only surface troll, 32 meters in average in 2019 and just 20 meters in 2020. Gillnetter is a very good addition since we will compare troll and gillnet catches, and definitely we will find something new. But all this just for surface. We did not see for the deeper layer, but we need to do. And actually we have opportunity to do that, not only by hydroacoustic. You know that it's a plan for Canadian vessel, for example, work just at daytime. So at night it's just a big opportunity to make for another hour, 130 minutes troll hull for 500 meters. And I think we will obtain a lot of surprise, which then can be connected to what we need to know about Pacific salmon in the upper layer. So likely that's all what I would like to say. Thank you.
Thank you, Vladimir. So I'm a couple options here. One, I can go open it up to the panel to say if they've got another thought that they wanted to add there. We can do that. But at the same time, I'm going to pose another question that actually is kind of near and dear to what we're going through right now and trying to raise funding for this. So, I mean, there's no question as we talk to these two agencies that what we're doing needs to be linked to a management outcome. In fact, we're in discussions right now, and if a lot of funding, and I think we heard that from Jim Murphy as well, that if this thing isn't driving an assessment for a commercial fishery, then it's going to be very hard to get funding. So, Jock White has put forward a question saying, if you could tell a funder one reason to fund the 22 expedition, what would you tell them? So I'm going to go back to the group again with that question and open it up that you can add if you'd like to add anything further in addition to what we heard from everybody on the first round. So I'll give you a second to think about that. What would you tell a funder one good reason to fund this expedition?
Well, Mark, I can start off.
Okay, hang on, Dick. So Brian beat you to the punch there. Then we'll go to Dick. Yeah, go ahead, Brian.
Well, we may have been going to say the same thing in the sense that, I mean, the reason that we have the resources now is that an industry supporter who recognizes the need to have a better understanding of what's determining returns and therefore the return to communities and industry, they want to understand what is the security of their investment. So the issue is not any single year or any single cruise. Their interest is in a longer-term understanding of the dynamics that would allow fishing industry in particular, and for fishing communities to have some confidence in the future, or are they going to have to adapt in some way? So, I mean, it sounds like it's kind of trite that it'll improve our understanding, but it's not just for that. It's to provide security for coastal communities that rely on fishing resources and for companies that support so many people throughout the coast. I mean, this is a billion-dollar industry throughout the North Pacific. And I don't think we should lose sight of the fact that our studies ultimately, what's really missing in the long term between the physical chemical oceanography is the links all the way through to fishing resources. And these are all the different species. So I don't think there's any question that there's value in what we're doing. It's just that we haven't done it in so long, it's going to be costly to catch up. That's the hard sell.
Mm-hmm. Thanks, Brian. Dick, you had thought?
I'll just add a little bit to what Brian said, because what Brian is telling you is what we have been telling industry. Now, here's something that's unfortunate, all right? What Jacques is saying, you could turn around and say, now what you have been doing and what people have been supporting for the past 30 years has not helped at all. The abundances are declining. You have no explanation for it. There is no understanding of what it is that is regulating the abundance. You can't build more hatcheries. Adding more juveniles to the ocean isn't working. So you have funders who have heard that this will improve abundances, okay? This will restore. This is the mantra, if you want, that everybody has been using for decades, and there's no question that it has not worked. What Riddle and I are doing is saying it hasn't worked because you don't see enough of the picture to see how what you're spending all your money on can actually fit into what I call optimizing or maximizing the ocean capacity to produce salmon. So the answer, unfortunately, is that we have to go to funders, who we have done, and they are smart guys, exactly what Brian said, and they understand that we really do not know how to manage salmon right now. That's clear to them. So they're not going to support the kind of work that has been going on. I'm not saying that's not useful, but I am saying much of what we do now is not going to give us the answers of what the production of salmon will be in a future of changing climate. So the answer to his question is that you have to think differently, and you have to recognize that there's a large component of knowledge that we don't have, and you can't really maximize current production until you have that. And that's what Riddle and I have been doing. And we have been able to get support from the industry because they actually recognize that there's information that we must have if they are going to understand what their future is.
So others that want to jump in, I won't go around the table, but others that thoughts. Lori, yeah.
Yeah, so I think similar thinking, but south of the border, we have a lot of populations of salmon that receive protection under the Endangered Species Act. And these are the same groups of fish, many of them, that we're catching out in the Gulf of Alaska. And I think it's really important, not so much from an economic sense, but also to understand what is going on, what the impacts when we change things in fresh water. You have to put the ocean part of the puzzle together to make sense of the fresh water. So you can't, if you do things in fresh water and then just say, oh yeah, we got a huge return last year, it must be what we did in fresh water. Well, actually, no, it's usually the ocean. And especially now when we're getting the just crazy swings, unexpected swings in populations, high and low, for being able to predict returns is not really working very well. I think it's really important, like both Dick and Brian said, to look beyond kind of the coastal area and understand what their true mechanisms are that are regulating salmon. And in this case, not for the economic sense of endangered species.
Thanks, Lori. Anybody else jump in? Vladimir?
Jackie was there.
Yeah. Oh, yeah, Vladimir, then Jackie.
Okay, thank you, Mark. Sorry, Jackie. Luckily, I have no some recipe to do that, since I still did not fundraise more than 10K from NPFC for the expedition. So I completely rely on opinions of Dick and Brian. But what I can add from my Western North Pacific experience, we can just tell them a history of Professor Shuntov, who started salmon research expeditions in early 1990s, where it was a very difficult time for Russian fishery science, almost no funding, no vessels, almost nothing. And it was just very lucky attempt, since Shuntov was confident that she will have a success. She tried to estimate an abundance of pink salmon in approaches to the coast. And it was happening that in that year, it was a very difficult situation. It was a lot of processing fleet gathered around the eastern Kamchatka. And at the mid-July, it was a hard situation when no fish, and fishermen started claim some research, and nobody can explain what happens. But after that, we received the first expedition's result, which shows that the pink are huge, but just delay in 2 weeks with approach. So it was a very good success that made a background for this research project for long years. So we can say now that if you will find a person who is so confident that he can develop such a big program to estimate the salmon abundance at high seas and then relate it to the further run, to the cost, and show that success. I think it also can attract attention and may bring some funds.
Thank you, Vladimir. Thoughts from others? Jackie, you had...
Yeah, I was going to say, I think forecasting and salmon management aside, what these two expeditions and funding the 2022 does is provides the observations of climate change. 2019 and 2020, one of the huge take-home slides for me was actually the slide of temperature at depth along Line P and Ocean Station Papa that Angelica and Tatiana provided for us in day 2 and illustrated it's just warm anomalies all the way down. So 2019 and 2020 was out there during many marine heat waves, but those are to increase in frequency and duration under climate change. And even if we anchor it to the few observations that we have in the past, and we saw some of those in terms of energy density or condition factor from the Pan-Pacific Japanese research vessels in the 1990s, things were a lot different, I think, maybe than what we were expecting. So the take-home message is that we're observing climate change, and what's the pressure being put on these fish by the driver through a whole bunch of factors, range from freshwater to coastal to these open oceans. And we're trying to build life cycle models, and instead of using proxies for the open ocean or their marine winter phase, we're actually making observations. And it's probably likely going to be that, industry is going to need to adapt. This is not going to change. And again, the way we've been managing has to change and adapt to climate change as well. And this could be a good anchor for illustrating some of the broad-reaching impacts on salmon, just beyond what we see in our nearshore freshwater.
Thanks, Jackie. Very good points. I think a related one.
Evgeny.
Oh, sorry, Evgeny, go ahead.
Well, I mean, if you wanted to say something, but if I can add, I think Vladimir already said something, that the program, which is reliably estimating on annual basis the open seas distribution, would be a very good selling point to the industry, because the relation, and even all those very small attempts which been done now by Dick and by Chris Neville, is that some of these numbers actually match up. That's already a very good indication that invest a few more years, and maybe there will be some equation coming up, very simple equation, which might be helpful. But what is also puzzling me is that whilst many fish stocks in the southern parts and are actually endangered already or declining quite dramatically, actually catches or productivity of the salmon overall in the North Pacific at the ever highest levels. It's the highest levels ever recorded, so it's right at the peak. I think there was lots of indications saying that, okay, we need to estimate carrying capacity, and apparently carrying capacity of how many salmon ocean can actually hold is different in the western and eastern parts, but fish migrating across, as we've learned as well. And I would say that maybe this historically high catches and historically high returns and productivity of the salmon is a result of maybe we should kind of rethink the concept. Maybe we should not put more hatchery fish into the ocean because northern parts actually benefiting from this, but the southern parts actually not. And I think that's something to think about, and we need more research for this.
Mm-hmm.
Mark,
Yeah,
can I
thanks,
follow up?
Evgeny. Sure. Yeah, go ahead.
Well, just a quick comment. I mean, I think
Yep.
we need to be careful not to be telling people what you've done in the past is wrong. What you've done in the past is not appropriate to the future. I mean, clearly, you've got to support what Jackie was saying. We're talking about a changing climate. We're talking about a changing environment that we have not confronted before, right? And so we really need to tackle it in a serious way so that we don't have a 20-year time lag. We can't afford that. So the urgency is that we know things are changing. What we did in the past is simply not appropriate to the future. The major hatchery idea is an issue that really is not likely to be productive or cost effective. And to Laurie's comment, I think we need to be very, very careful to make sure that we link the full life cycle. I think it was Jackie that said that again, that what you do in freshwater habitats and restoration and management is still extremely important to the future of salmon, even with the changes in the ocean. So we cannot break that link. We just have to make the tie more clear and what's the appropriate actions.
Mm-hmm. Yeah, excellent points. I just want to open it up to the audience if they want to comment. We've got a number of comments in there. And a related one that I'll start with is from Urawa-san. And I guess this speaks to both Jackie and Laurie were speaking about, and Jim, relating to juvenile surveys and this idea that we need some end-to-end models, life history models, and so Urawa-san is saying energy content level prior to winter season may affect the survival of overwintering salmon. We need follow-up research before the 2022 winter expedition. I expect the Japanese summer survey in the Bering Sea can provide such data, and I hope all other summer and fall regular high seas surveys consider this aspect, this idea of linking. So I don't know if we need to comment on that or, yeah. I heard somebody.
Well, I don't know of another survey. Well,
I
what
guess
other
I'm
survey
thinking
is there?
about. Well, I guess we've talked to Laurie and Jackie and others about their coastal surveys of juveniles. So I think we may have been messed up, but I think COVID disrupted and funding has disrupted a number of those juvenile surveys that might line up with seeing those cohorts when they're at sea in winter 2022. But I think connecting the Bering Sea, but also these coastal surveys would be important. I don't know, Laurie, do you have I know you've given some thought to this idea.
Yeah, I think it's really important to, follow these same groups of fish as they're in coastal areas out into the high seas, and then actually returning as adults to compare, which ones, a lot like Christoph did with his genomic work, looking at pathogens, et cetera, where you can see which fish appear to be falling out, what's there from, I think, these really critical 3 stages, which is the coastal area, the high seas, and then back to the adults, to really understand what's going on.
Right. They've got Gennady has got a comment that full North Pacific scale is the right idea. The southern edge of the survey is a question. We need more remote sensing. We're in an ocean region with million years story, and at the very slow upwelling ocean scale zone, and I'm losing it, with redistribution currents. So I think, that has been brought up before. I think it was Jackie, making sure we cover the full distribution, and clearly that's where were the pinks, and that's southern end. And I guess I'll inject a comment there. I think I saw some presentations of historic data over the last 3 days that I was very impressed with, and we're doing some work on data recovery of data that high seas data that are at risk of being lost. And I think this historic data, we need to mine it in more detail and try to make sure we nail that southern end of the distribution that we're trying to hit here. I don't know if anybody else has anything in that area that they want to respond to.
Well, I mean, in the same
Can I?
vein, Mark, Lexi, I just finished. I was just going to say that if you're going to deal with historical data, my God, you got to go to Russia. You've got to get whether it's translated or not. I mean, Shuntov has a spectacular career in in oceanography, biological production. So we have to deal with the Russians and integrate these data sets. And Japan, of course, Japan with years and years of high seas research that we have to capture.
Yeah, absolutely agree. And by the way, when Vladimir Yurchenko was speaking, Vyacheslav Petrovich Shuntov stood nearby and he said hello to every one of you. Moreover, so to address the previous point, so I would like to emphasize that in addition to ecological insights which 2022 expedition can provide us, so in my presentation and as Jim Murphy also showed that a really well organized high sea survey can significantly reduce a bias and uncertainty in the salmon forecast, and it would have a really good perspectives for fishermen. Also, Dick Beamish mentioned that he is going to organize a gillnet and longline survey, and I think that it is a very good idea because if the stations will be at the same place with the troll vessel, we will get a real direct comparison of between these two types of gear, and this way we can standardize data which was previously get for 50 years, for almost 50 years, and it will give us a good perspective in terms. So we will be able to compare 2022 data with previous studies.
Thank you, Alexei. So another one from Jock White saying, I think it was bringing forward from earlier, are coho being sampled by trawls with equal efficiency during day and night? Sockeye as well, question mark. If these species are vertically migrating, why? And are we sampling macro zooplankton and micronecton effectively? Do we need to employ a 1 or a 3 meter mock nest in 2022? I think I'll let Jackie answer some of that because she is deploying a mock nest in 2022.
Yeah, I love the mock nest. So that was one of the first things I wanted to do. So myself and Stefan Gauthier and Akash Sastry, along with John Dower at UVic, are going to be working on that acoustic project that Mark touched upon, and we'll be deploying a Mockness gear to sort of fill in that gap. So we'll have the acoustics from underway, and hopefully some of the target signals will be verified, well, obviously with bongos and in the TrawlNet, but also the Mocknet. So yes, we are going to be doing that. Great. I think, and we've heard over and over again, right? It's the missing piece in terms of the trophic food link.
Right.
So it's going to be awesome.
Evgeny, you got your hand up.
Jackie, you can finish if you want.
I think everybody can agree Mockness is going to be awesome.
Yes, I just wanted to add to this, and as far as I understand, in Canada we have only Mockness 1, which would be good enough. However, if there is actually any opportunity to deploy Mockness 3 meter, which is a very cumbersome gear, I've worked with this, and it's quite something. It's much more difficult to deploy than actually a huge midwater trawl. But if there is opportunity, that would be a proper micronecton macrozooplankton gear catching this very well. Everything from, let's say, jellyfish to squid. It would be awesome to do this.
Lori.
I think Jack brings up a really interesting question, though, because we did see really big differences in the catches of the different salmon species day and night. So we caught most of our chum salmon during the day, and we caught most of the night and cope at night. And the question is, where are they at other times when you're not catching them in the surface? Where are they? Obviously, presumably deeper, but how deep? And I think that's a really interesting question. And kind of what are they doing, and why is it so different between chum and these other two species? That I think kind of dialing it a little bit to look at things like diets, day versus night, etc., would be very interesting, as well as trying to do some deeper tows, either during the day or at night, and how deep are they, as well as using the acoustics to see where you think they are. So it is a really intriguing question that I don't think we have a very good answer for.
Yeah, Brian?
Well, I was just going to say right along that line, I mean, we can fish all we want, but one of the new technologies are these archival tags, and they're getting smaller, but they're expensive. So I think we need in our thinking to, if there are key questions to ask, let's see if there's some new technologies, and maybe there's some really bright people that can figure out how to miniaturize them. Even if we reduce the battery life from 2 months to a month or 2 weeks, you could learn a huge amount by getting some tags on and letting the animal use its environment.
Yeah, I know, Jackie, you were proposing we should be applying some tags as well. And it's something we kind of pulled back from after 2019, but I think it is something we need to be thinking about. I agree, Brian.
I think the big challenge with a different, a different technique than the live box, as far as I understand. That was really difficult and didn't really work out.
Yeah, I was going to say that
young people with different ideas.
Long lines, old people with old ideas.
Hook and line is definitely a good way.
Yeah.
Yeah, I'll sign up for that, the hook and line. We've got we've got one more here. Brian Hunt, large scale is important, but we still lack information on individual behavior of salmon, example, vertical distribution and interaction with changing ocean conditions, oxycline, thermocline, etc. Eddies are intriguing and are so abundant in the Gulf of Alaska. How do salmon use them? How important are they for production? Yeah, I was certainly struck by what we saw from the oceanography as well. Jock was just, I think, closing out on our conversation earlier. Would it be fair to say funders that understanding the behavior and responses of specific salmon populations in response to emerging ocean conditions can help us manage both hatchery and wild populations where we have more control in freshwater systems going forward? I think that the legacy effects and of management behaviors in levers that we have need to be informed by the ocean. So I think we've made that case pretty strongly.
Hang on a minute. Does more control imply that you can improve abundances? Is that what more control means? Because of course that's not correct. If the ocean carrying capacity, if the coastal capacity for salmon is declining, and you believe in ecology, then the production in fresh water is limited, period. So you can have more control, but it doesn't mean that the more control is going to give you more salmon. And if you then tell, a caring public and funders that by providing more support for that kind of research, they can expect more returns, then you're misleading the public when the coastal capacity for salmon is declining.
I think I think it's making more better decisions about how to utilize your freshwater, tactics in relation to. You need to understand what's going on in the ocean. I think that's
Mark,
what I
you can
read
say
in what
that.
he
Hang
said
on,
there.
that's a nice thing to say. All right, but what we're looking at is really trying to understand something that's almost an emergency. So you want the general public to understand that there's information that we simply must have if we're going to be what I call professional stewards. So you can try to make everybody feel good, right? Which of course is nice, but you also have to recognize that if the coastal capacity to produce salmon is declining, then the freshwater side of it, adding more juveniles to the ocean, ain't going to work.
Mm-hmm. Okay, we are coming up to the end of our time, and I'm just going to say a few thank yous here, and then I'm going to pass it back to you, Brian, to close it up. I'd just like to thank the panel today for at the end of 3 days, and it's a lot of energy to be on Zoom for that long, and I thank everybody for their passion and the ideas. I think I'm coming away with this inspired. The science, my head is ready to explode in terms of trying to assimilate what we've learned, but I'm incredibly excited about what's been learned, and sort of gets me motivated to keep pushing on this where we need to go in terms of the fundraising to bring it home in terms of getting out there in 2022. So thanks to Oh Boy Productions for managing Herding Us Cats in this, and to the team and Dick and Brian and all their work. So thank you very much, and thanks to the audience for being there with us. I'll pass it back to you, Brian.
Okay, thanks, Mark. Well, I mean, obviously you've said what I need to say in terms of thanking Oh Boy for taking on this challenge, and certainly want to thank all the people. We've had excellent participation. My first day count was around 140. Yesterday was about 110. It obviously has been a success in reaching out to people. So we thank everybody that participated for their interest. You're certainly welcome to send me any notes if you have additional ideas and thoughts. And this topic is certainly not going away. We have the March 2022 coming up. We still have challenges to put that in place and that, but the interest is there and we'll proceed. And don't forget that after that, I think that's the real issue, is how we build a big multinational, multi-year program that really links climate to oceanography to fisheries. I'm always impressed with how much detail and information people have about climate effects and physical and chemical elements of the oceanography, but there's not as many impressive effects in terms of linking that through to fish and to communities that rely on fisheries, etc. So this has been great. Dick and I will proceed. We're doing that. Jackie's involved in Canada. So I think this has been very useful. I just want to say thanks to everybody, and particularly to Northern BC's Northern Development and Innovation Fund for supporting this, and for supporting the publication of this material through NPAFC. So thanks very much. And Dick may want to actually have a comment at the end as well. It was his idea to push forward with the virtual.
The same. Well, you've all said it. I think it worked really well. The presentations were extremely well done, and I think I don't know who's still listening to this, but this is a volunteer effort. The people that presented, some of them are employed, and maybe that their employer paid for some of this, but in general, all of us that are participating in this is the two expeditions and all of the arrangements. Just about everybody in some way is giving up some of their time. So I think it's a really nice effort, and the people that have supported the expeditions, and as Brian mentioned, the Northern Development Fund, but particularly all of you guys that are here now and we're participating in the 3 days, and the people that prepared papers. I think that says a lot about everybody's passion for understanding what is regulating salmon abundance. So it worked out fine, except for a few glitches that I had, but that'll always happen.